indeed a very difficult phase for all of us, and particularly for those engaged in imparting education. Especially when the institutions like ours is situated in a remote Adivasi belt with poor internet connectivity and erratic power supply, the challenges are even more acute and unsurmountable. However, every obstacle is in itself a potential opportunity. The offline classes gave way to the online techniques, and despite the many infrastructural glitches, we as a team were able to surmount most of the difficulties and in fact seek newer avenues to improve our teaching learning performance. One of the ways to do this was to organize a conference on an international scale, which would enable experts in the field spread over the world to come together on this platform and present, discuss innovative ideas and projects. In 2021-22, we arranged the first international conference. The present one is the second in the series, and we look forward to arranging similar conferences annually and, if possible, even in person. Support of the teaching fraternity and enthusiasm of our staff and students would certainly help us in making international conferences a regular annual feature of our college. Our college was established in 1994 with the help of a munificent donation given by our primary school teacher, late Sri Nagajibai Mehta. He served in our Gokhale Education Society's SPH High School, which is a 101 years old institution. Gokhale Education Society, with its motto, above self, above politics, has led a silent transformation and contributed to nation building from 1919, right from its inception. Inspired by visionaries like Principal T.A. Kulkarni, Acharya Bhise, Chitre Guruji, and Save Sir, and led by the present office bearers, Chairman Principal S.B. Pandit, Secretary and Director General Sir Dr. M.S. Musavi, Director HR Dr. Mrs. D.P. Deshpande. The Gokhale Education Society manages about 140 institutions across 3 zones, Nasik, Mumbai and Palghar in Maharashtra. It has brought the elixir of education, the economically and socially marginalized Adivasi community of the Palghar district, erstwhile Thane, and has proved a boon for especially the girl students who could never have moved outside the village to pursue higher studies. Our college, which began as a single faculty science college, permanently affiliated to the Mumbai University, has over the past 26 years, developed into a science and a commerce college with facilities for graduate and postgraduate studies in physics, chemistry, information technology, computer science, biotechnology, microbiology, botany, and zoology. And in commerce, Bachelor of Commerce, Masters in Commerce, and Bachelor in Finance and Accounting. Our college received the Best College Award Rural in 2012 from Mumbai University and a Maharashtra State Award for Dagal Zaniwanta in 2014. Today, we have research facilities in the physics, botany, and zoology departments leading to PhD. And we have guides, recognized guides for the PhD program. The geographical situatedness, economic backwardness of the community at large does pose important problems. But every coin has 2 sides to it, the flop and the flip side. The flip side of the institution is its pollution-free, academic-friendly environment, both naturally and the teaching learning environment. I could go on describing the strengths of my institution, but the most significant is the cooperative spirit and the strong bond of solidarity we as an N.V. Mehta College family share among ourselves. Today we look forward to in-depth discussions and deliberations by eminent speakers and delegates, as inclusivity and research are the 2 major pillars of our institution as much as they are part of the New Education Policy 2020. I am truly happy to be able to speak so highly of my institution and delighted to welcome you all today's inauguration session. I'm sure the conference, with the help of all of you, will be a huge success. Your support and good wishes. Thank you.
Madam, it gives me an immense pleasure to introduce chief guest for today's function, Professor Dr. Hiren Doshi sir. He is an extrovert and innovative professor in chemistry, aptitude for research and 20 years professional experience as an academician and researcher in chemistry at educational institutes and industries in India. He is presently serving as Pro Vice Chancellor, Sage University, Indore. Sir has served at ARI BS, Anand, Gujarat for about 8 years in administrative position. Sir has worked at Rubamin Pharma Limited in research fields for about 4 years. Sir has also worked with Zydus Cadila, Baroda as a senior research scientist. Sir has a huge experience for postgraduate teaching in MSc chemistry program. Sir has guided PhD students, and 2 students have been awarded their PhD degrees, and 8 more students are pursuing PhD under Sir's guidance. Sir has a strong research background and had published more than around 30 research articles having high impact factor index Thomson and Reuters in reputed international journals. Sir has published number of book chapters with reputed publication house Taylor and Francis. Sir has won international patent and 7 Indian patents. Sir's field of interest are medicinal chemistry, drug design, bioremediation, and now I request Professor Dr. Hiren Doshi sir to guide us. Over to you, sir.
Thank you, Banjarji. Good morning, everyone. I'm audible?
Yes, sir.
Okay. Respected Director General, Secretary MS Gosavi, Principal Dr. A.S. Kulkarni ma'am, Convener NT Naragunde, Head of the Department Chemistry, Professor P.K. Gugari, Vice Principal and Head Department of Botany, GES, eminent speaker like Professor Satya Suvarnaji, and dignifying all dignitaries, staff members, and my dear friends. Good morning to one and all present here. It is giving me an immense pleasure to address all of you in opening ceremony in this morning to this international conference. It is an honor for me to hear among the scholars, researchers, teachers, students. I would like to thank the organizer for inviting me today's function as a chief guest in today's international virtual conference on recent advances in chemical and biological sciences at the NB Mehta Science and Commerce College of Gokhale Education Society, Bordi, Palghar districts. It is commendable that this college located in the remote tribal Adivasi belt of the Palghar districts has taken this initiative for international conference. Science is a life, and life is a full of science. This is the simple sentence, but it is embedded in itself all the different type of laws, chemical reactions, theories, calculations, phenomena, and everything that are required for life to exist on Earth. Chemical science and biological science are two very important part of interestingly these are the two sides of the coin. Both are related and dependent on each other. The question how life organized on Earth is still unanswered convincingly. Chemistry has a role play with the handshaking of environment. We are the race against time of smarter development. We are using science to build a future we want. The conference topic is extremely timely, especially given the world's constructive struggle to post-pandemic devastation. The construction work will not be possible without the help of collaboration of people from all walks of life. The only way to overcome the enormous challenges posed by COVID-19 and its subsequent strain is through an smart interdisciplinary approach and sustained efforts towards teamwork in all aspects of life. Most importantly, the approach will also keep in view sustainable development. We have entered an era in which we are all concerned with the well-being of others, both now and in the future. There are premises that underpin the conference theme and sub-themes. Scientific thought and scientific methodology are our most important collective effort in elevating human distress while protecting entire ecosystem. The conference focuses on two very interesting fields in the field of chemical and biological science. Anyone can visualize the several upcoming developments that are emerging with which will change the face of Earth. Few developments are like artificial humic matter from biomass. This synthetic matter will improve crop productivity. Blockchain technology, an important process that has been adopted by chemical companies. New imaging light and chemiluminescence technologies, layer at living light, chemical synthetic of genetic material, a huge possibility, supervetability, a mixed material science, biological and chemical science with huge potential in day-to-day life, artificial organs, 3D printing or printed organs, genetic engineering for gene modifications. These are the few important glittering technologies that will have huge impact with increasing investment in research in regard with GDP also. The accountability and adaptability has increased. Such important conference offer a platform to young students, young researchers, scholars to exchange idea and learn from the expertise to top-notch scientists from across the world. Saying that exchange of knowledge is a key to achieve academic excellence. Professor Nilofar underlined the need for academic institutions to shun working in isolation and encourage the student and faculty exchange program to solve pressing the social issue through multidisciplinary and interdisciplinary research. This thought is made true which such a remote college disseminating knowledge by bringing in an international conference with delegates of international and national reputes. Biological science represent an interdisciplinary science combining various areas of research in medicine, modern biology, combining them with advanced method of analysis. The topic can help teachers, researchers, and students as it will initiate discussion on new and emerging areas, offer a platform for exchange, new ideas and strategies. This will also inspire more students to undertake research which proves beneficial for all. Getting to know a large number of researchers bring countless advantages. A conference of this kind will help to establish an academic rapport globally. Eminent scholars and aspiring young researchers will imbibe in the knowledge that they acquire. The college is situated in the tribal belt of Palghar districts. Its remoteness may sometimes be boon as may natural avenues are left untouched and therefore unhampered, as already Kulkarni ma'am said. This conference can help to create interest in topics and help in enhancing our knowledge regarding indigenous knowledge of biological and chemical science necessary for the well-being communities. There is a quote of Rigveda, Ano Badra Pratava Yantu Viswar, means let noble thoughts come to us from all and every direction in the universe. I would like to thank organizer for giving me an opportunity to put my words in the international conference on the topic common concern, and I wish a grand success. I hope that deliberation of this occasion will useful for policymaker in the era of science. I also special thanks to PK Borisab who invite me in this conference. I wish the committee all the very best. Thank you. Thank you very much.
Thank you.
Thank you, sir, for your inspiring speech. President of today's function, Secretary to Gokhale Education Society, President of MSG Foundation, Director of Bitco Institute of Management Studies and Research, Sir Dr. M.S. Gosavi, sir, is a dynamic personality, spellbound speaker, and outstanding management scientist. He has served as a chairman, Business Administration Board, Pune, and President of Indian Commerce Association. Sir was the chairperson at All Asia Management Conference in Tokyo and President of Indian Association of Management Development. Sir Dr. Gosavi, sir, is a recipient of Silver Memento Award by the hands of the Governor of Maharashtra, International Award for Making University Education Entrepreneurship, Rajiv Gandhi Foundation Peace Award, Jnana Hira Award, Master Teacher of the Millennium Award. Sir was selected as Man of the Year by International Biographical Association, USA in 2002 and World Education Fellow, Tasmania in 2000. Sir Dr. M.S. Gosavi, sir, has been awarded with Knight of the Millennium Sir title at the hands of the Sheriff, Mumbai in 2000. Sir has authored 20 books and more than 100 research papers and edited 100 books. He has received Nashik Bhushan and Fulton Bhushan Award. Sir Gosavi, sir, is a firm believer in building human capital through imparting quality education. I request Sir Dr. Gosavi, sir, to deliver presidential speech. Sir Gosavi, sir.
You are not audible, sir.
Video is not audible. They are working on it, sir.
Just a moment, sir.
Yeah.
Bordi, as you all know, is a small village. It's a gram panchayat having a population of 5,000 people. It was as early as in 1920 that Principal T.A. thought of starting a agricultural school in this area for the benefit of Adivasi children. And this was the first attempt to provide education to Adivasi in the whole of Asia. The institution has delivered quality education because of the teachers which the society attracted at that time and the way in which education was imparted.
There are some technical glitches.
And self-identity. The topic of research
Please bear with us.
is recent advances in biological and chemical sciences. In fact, The society has great rich center. The society has great pleasure in welcoming the distinguished guests from different parts of the world for the international conference organized by the Bordi College at Bordi or Bordi. I am very happy that the distinguished guests have made it a point. The society has great pleasure in welcoming the distinguished guests from different parts of the world for the international conference organized by the Bordi College at Bordi or Bordi. I am very happy that the distinguished guests have made it a point to send their addresses and participate in this conference and technical sessions, which will go a long way in enhancing the quality of life. Bordi, as you all know, is a small village. It's a gram panchayat. Having a population of 1,000 people. It was as early as in 1920 that Principal T.A. thought of starting an agricultural school in this area for the benefit of Adivasi.
We are extremely sorry for the complex. Just bear with us for a few minutes.
To provide education to Adivasis in the whole of Asia. The institution has delivered quality education because of the teachers which the society attracted at that time and the way in which education was imparted with selflessness and self-identity. The topic of research which is introduced in the conference is recent advances in biological and chemical sciences. In fact, biosciences have come to play a very important role in the building up of India in the present time. This is an entirely agricultural part of Maharashtra, where population is available for increasing productivity on the basis of methods of cultivation to be adopted by them in a meaningful way. So our society did not only start a school and started educational program as such, but our society made an effort to depute certain teachers from this particular area to different countries in the world like Japan, Philippines, Australia to find out how best varieties of agricultural products and agricultural seeds can be obtained. And I'm very happy to say that they have specialized in the field of grass. The Clay Education Society is known for grass management as such, making the producer, the farmer, an entrepreneur. Here the problem of water was not there. The problem of labor was not there. The problem was only how to have enlightened citizens and people so that they will take up to the modern methods of cultivation. So Dr. Patil, who was sent to Japan for not less than 6 months to become a farmer there in the farm, came back to India with high research and introduced Japanese method of cultivation in this area for the first time in India. As a result of which, not only the grass seed which was created in Japan was tried in India, but because of the various combinations which were put under practical experimentation, our center at Kosba in this area gave a new seed in grass cultivation. It is known as dasar grass. It is a grass seed which produces grass 12 times once the water is given to the seed. And you may have as many as 12 or 14 cuttings, and the milk yielding capacity of the cattle goes on increasing as the cattle gets this type of grass. So this is a unique contribution of Kosba Center, the Vocational Education Society, to the world of agriculture, where milk producing capacity of the cattle was increased without increasing the cost of either raw material or fodder or even for maintaining the cattle. So it was from that point of view that this experiment was made very successful. Another thing was in-depth cultivation method was adopted so that little less ground which is available or land which is available was put to use, which could produce more grain. And therefore this particular center expanded like anything in those days. And in the last 100 years, our school has completed 100 years now. We established a higher education center in 1994 with all types of sciences to be taught to the students for making them more and more scientifically sound and also all-round personality. It was Mr. Mehta who came to our rescue in those days. In the 75th year of the Bordi Center, when we wanted to felicitate some of our outstanding past students and some of our eminent past teachers, we found Mr. Mehta as one of the persons available who was a teacher in this particular center for a very long time. And he retired in 1969. At that time, he said that if I am allowed to work for a little more time, I will be very happy. And government of Maharashtra at that time, being a granted aid institution, was very careful in saying that all cases of retirement should be considered on the same date, and new person should be given an opportunity to get the job. But society on its own thought that Nagarji Mehta, who is coming from different parts of Maharashtra and also from Gujarat, had some ideas of development of sciences in agricultural area. Therefore, we gave him extension for 1 year. And like that, for 6 years he was extension given. And he produced, with the help of his family, new methods of cultivation in different areas, along with the subject which he taught there. So this was, a unique opportunity for the society to make a mark in this particular area of biosciences. By that time, Nashik also has come out with postgraduate programs and many new sciences. Computer science has come as one of the subjects, and this has also given great fillip to the educational pattern at the postgraduate level. Therefore, in the biosciences, HPT College started postgraduate courses. It was linked with Reward Institution when the college was started in 1992. And today it has become a rich center, giving enrichment to the entire society there in the faculty of commerce and in the faculty of science. The recent trends which have come up in these sciences are actually, you see, making the agricultural production more and more tasty and consumable, and the price of the product is going down. It is from that point of view that these modifications which are to be made in the cultivation pattern, in the seeds to be used, and in the method of using, helping the labor to reduce the efforts involved in this particular program, which is called as integrated farming, integrated economic development, has come up only through this type of cultivation. So all the problems relating to agriculture and farming, particularly soil, testing of soil. Number 2 is rearing of cattle. Number 3 is training to the laborer. And number 4 is conservation of water. So all these problems which are to be tackled properly require modern touch. And therefore recent advances in these sciences have helped agriculture in different parts of the country, particularly in India, making whole production more useful and content-oriented. Proteins has been called from the grains which we are producing is very important. It is pH 7.4, which is maintained in the body of a person only by this method of agricultural cultivation. And therefore, all products which have been produced here, particularly horticultural products, there are several horticultural trees which have come up in this region. And chiku is one such product which has come to India in Maharashtra for the first time from Parsi area, from Kathiawar, and Local Education Society, which is making that particular product popular in those days. We are the first to produce chikus in India. We are first to produce, quality chiku and exporting it outside. And in addition to that, the bananas and the mangoes, these trees also flourish in this particular area. It was told to us when we started this particular program that rice cannot be cultivated in this area. It will not come. But we have made an experiment. Now it is also proved that in this area, which is very hilly and mountainous and terrain land, where all the water of rain which comes through the clouds goes ultimately immediately to the sea, which is very near at a distance of 4.5 kilometers. So there is no staggering of water in this particular piece of land that we are having. We have 300 acres of land. We are doing agriculture for the last 100 years through all these methods. And I am very happy that Anudaiva and Tarabai Model, who are also the originators of the children education in India, have helped us by establishing their center near our farm so that all children of Adivasis were given quality education right from pre-primary stage. Primary stage. We had a school there right from 1920, which has also flourished. And we established college there in 1994, Science and Commerce Wings, to postgraduate level, affiliated to University of Bombay. As a result of which, Adivasi people who were otherwise torn off from different areas. They cannot go to places like Thana or Ratnagiri or Kolhapur or Bombay for higher education, could easily take higher education. And particularly the girls who are not going beyond 10th standard have completed the education at postgraduate level without any cost estimate. So in these sciences, basic education is to be provided, which we have provided. And these recent advances in these sciences, which are there right from the days of Radikal, he is the first person in the field of chemistry to give us advances in the chemical field. And we have also many teachers like Vijapurkar, who have made contribution in the field of biological sciences. And today all science was established. Till this day, thousands of students have gone from the portals of and also in other sciences, electrification and electronics, etc. From the bottom of our heart. And I am sure that the proceedings of this conference will go a long way in making ourselves more enlightened for the new problems and challenges we are facing at present in the field of agriculture. Thank you very much.
Thank you, sir. Firstly, I would like to apologize for the poor sound quality. There were some network issues that we are facing just now. I'm extremely sorry for that. But thank you, sir, for your motivational words. You are always inspiring to us. Thank you once again.
Now I request Chairman of this program, Head of the Chemistry Department, Dr. Nilgude sir, to give vote of thanks. Over to you.
Thank you, Benazir. Good morning, everyone. It is honor for me to be a part of this wonderful event of 2 days international conference. I am very glad to be standing here to express my vote of thanks on behalf of staff and students of NP Mehta Science College and Commerce College Bordi. Honorable Chief Guest Professor Hiren Doshi, Pro Vice Chancellor, Sales University Indore, respected Sir M.S. Gosavi, Director General of Gokhale Education Society Nasik, respected Principal Dr. Kulkarni Madam, Vice Principal Dr. Bordi Sir, Vice Principal and Convener of this conference Professor Pankaj Gogari Sir, teachers, participants, paper presenters, my colleagues and dear students. I would like to extend my heartfelt gratitude to today's Chief Guest Professor Hiren Doshi for accepting our invitation despite his busy schedule. We thank for your guidance to our students and participants of this conference. I would like to extend my gratitude to our Director General, Sir M.S. Gosavi, for presidential address. Sir, we are all inspired by your valuable and inspiring words. I sincerely thank to our college principal, Dr. Kulkarni Madam, for her guidance and continuous support in organizing this conference. I also extend my thanks to our Vice Principal, Dr. P.M. Bordi Sir, Vice Principal, Professor Pankaj Gogari, for their valuable support. I would like to thank all paper presenters, participants, my colleagues, and dear students, and look forward to your active participation in the conference. An event like this cannot happen overnight. It all started rolling weeks ago. It requires planning and bird's eyes for details. We have been fortunate enough to be backed by a team of very motivated and dedicated colleagues from chemistry, botany, zoology, microbiology, and biochemistry department. I would like to thank Mr. Amit Raut for extending helping hand in technical support. Last but not the least, I thank to all those who have helped us directly or indirectly in organizing this today's international conference. Thank you very much. Over to Benazir.
Thank you, sir. Thank you for your vote of thanks. The keynote speaker for today's conference is Professor Satya Putan Vettil, Associate Professor, Department of Neuroscience. I welcome you, sir, for the conference. We have with us Daniel Kulangara, who is Assistant Professor, Howard College, Florida. I request Mr. Kulangara, sir, to introduce our keynote speaker for today. Over to you, Mr. Kulangara, sir.
Good morning, all of you.
Good morning, sir.
First of all, I would like to thank Dr. Pankaj Gogri, Vice Principal and Department Head of Botany. He was my student a couple of years back, and I'm so happy to see him, and thank you for inviting me. And it's my great pleasure and honor to introduce the keynote speaker for today, Dr. Satya Putan Vettil. When we had a conference in our college, Dr. Satya was kind enough to come and deliver a speech on biotechnology, and I still remember his presentation. Dr. Satya Putan Vettil is currently a tenured professor in the Department of Neuroscience at the University of Florida Scripps Biomedical Research. His lab is interested in the mechanisms of long-term memory, storage, and Alzheimer's disease. He is also actively engaged in developing therapeutics for Alzheimer's disease. His lab is currently funded by National Institute of Health and NSF and provide foundations such as Alzheimer's Drug Discovery Foundation. His lab is known for the discovery of the role of axonal transport in memory storage and the significance of long non-coding RNAs in modulating synapse function. He was born in Palakkad, Kerala, where he did his undergraduate degree in BSc Agricultural Sciences, then took MSc in Agricultural Biotechnology from Tamil Nadu Agriculture University. He then worked briefly at ICGEB, New Delhi, and RICE Genome Program in Japan. He received a PhD in biochemistry from Washington State University and postdoctoral training from Nobel laureate Eric Kandel at Columbia University. He set up his own lab at Scripps Research, which is close to our place. The county is Palm Beach County and in the state of Florida. He has received multiple awards such as the Whitehall Award and a Career Award from National Science. And as you all know, in Florida, it is almost 1 o'clock in the night. And really, I thank Dr. Satya for accepting the invitation. Over to Dr. Satya.
Thank you. Good morning to you all. I want to begin by, hearing all these things from Principal and and Dr. Doshi about, how beautiful the campus is, and the struggles that you are facing to get this up and running. At the same time, organizing such an international meeting. This is kind of an inspiring story, to bringing people together, irrespective of where you are, using this latest technology so that we can share ideas and learn from each other. So congratulations to the organizers for setting up this kind of conference. I'm sure 100 years from now we will be meeting from different planets, because the technology we may be able to bring together, and we should be able to meet as often as required. So congratulations again, and very best for many more such fantastic conferences to happen in the future. I wanted to thank Daniel Kulangara. He's been, one of my friends for a number of years now. I mean, for giving me recommending me and giving me this opportunity to uh, give this keynote address. I wanted to accomplish a few things during my talk. First point is, something like something that Professor Doshi was mentioning about the advances in techniques, in different methodologies, kind of changing our lives. He kind of very aptly put um, one of the most recent advances that we all are actually enjoying, those of us who received the COVID vaccine. This is basically an RNA-based vaccine. This involves, the ribonucleic acid you inject into your muscle. It produces, it's going to protect you from number of other infections. So this is kind of a technology that's, revolutionizing our lives, and you're going to see a lot more of these things. You go to an hospital and you probably don't need to go to the hospital ever again for years. So that kind of giving a kind of a nice transition to what I wanted to talk to you all today. This is about a species of RNA called the long non-coding RNA. And I'm going to take you through a bit more in detail. So that is the scientific part. And the second aspect I wanted to show you is that, I want to share with you some real science data that is not published. This is basically to inspire students to see, how certain things are done. We can have a story, but we wanted to know, how actually things are made. So you will see a story that's being developed, identifying a simple RNA molecule to trying to find its function in the brain. So basically I'm a neuroscientist, and so my lab is actively trying to understand, higher brain functions. So with this, I'm going to begin my presentation. I hope it's going to work.
Yes, sir, your screen is there. We can see your screen. Sir, take your own time. No worries. Share as a full screen.
Sir, the screen is now visible.
Screen is visible.
Hello, Satya sir.
Satya sir, please unmute yourself.
Hello, Amit Bhau. Satya sir's
Yes,
mic
sir.
is not working. Satya sir mic is not working. He is not able to find the mic sign. So he is
Okay,
requested to
sir.
run video. He is requested to run the video.
Okay. Okay,
Recording
sir.
video.
We will do that. Just one.
Okay.
Yeah. Just
Okay.
one
No
minute, sir.
problem. No.
Yeah. We'll present a recording.
Okay, so we will run recorded video. No worries. Now you are audible. Now you are audible.
Have my audio going or my video going?
No, it's all right,
I can
sir.
just
We'll
show
run.
my screen. The screen is not moving for me. I'm really sorry about it, but
No,
I'm
no.
happy to take you through the contents without the
It's
slides, and if that is okay
no,
with you, or we have a recorded version of it.
sir. We will run a recorded video, and then you can quench the question answer.
Sir, can we play the recorded video? Dr. Satya? Professor Satya? Professor Satya, could we play the recorded video? Is it okay?
I can't hear anything either now.
Yes, permitted. Just run the video. Run the
Okay, sir.
video.
Okay, sir.
That I am standing right in front of you and giving this talk in person. I'm on the other side of the globe, so please accept my apologies. I wish the very best for the success of this wonderful meeting. And I'm sure the students and the teachers and the scientists, we all will have a lot of opportunities to interact, make connections, and and learn something new. And that's the ultimate goal of this kind of meeting. So just to keep inspiring each other. I want to begin by thanking one of my long-term friends, Dr. Daniel Klingera, and members of this advisory committee for giving me this fantastic opportunity to share some of the work going on in my lab. As you all know from the title, that we are interested in a species of ribonucleic acids called the long non-coding RNAs, especially their functions in the brain and in mediating, setting up long-term memories. The importance of the RNA is nothing new. For example, like in the most recent advance in the field is from the field of RNA therapeutics. For example, a few years ago, almost 2 years ago, when the COVID was spreading like a pandemic and shaking our lives very badly, the RNA-based vaccine was developed in a record time, and that kind of changed the course of this pandemic. Just to give an example, like how versatile this RNA-based technologies are, and we all can expect a lot of similar new medicines coming up from this RNA research. What I'm going to talk to you is mostly about this RNA in mammalian brain, but I want to begin by giving you all a brief introduction to human brain. Human brain is one of the most extraordinarily complex organs known to us. It consists of about 80 billion neurons as well as similar 80 billion non-neuronal cells. And something very peculiar about the neurons is that they connect to each other in specific manner. Each neuron is able to talk to 10,000 other neurons, making 10,000 synaptic connections. Consider the magnitude of this number. For example, the number of stars in the Milky Way is estimated to be about 100 billion, whereas the number of connections in each of our brain is about 1 trillion connections. So that's a complex system, complex things going on. A major challenge for the neuroscientists are facing these days is to make sense of these connections and to see that what happened to these connections when we learn, when we remember something new, and also when we are exposed to different diseases and during aging and in neurodegenerative disorders such as Alzheimer's and Parkinson's disease. Brain is about 3 pounds of jelly. Brain is about 3 pounds of jellies, like the tissue. It's kind of organized into different compartments such as cerebrum, cerebellum, brainstem, things like that, but you don't actually see these colors when we open our skull and look at the brain. But if you use some of these some of these modern techniques such as the diffusion tensor imaging, you tend to see these kind of long distance connections in the brain. For example, here the neuron is connected from this anterior to the posterior, as well as the dorsal to the ventral. You can see that these kind of this is called the long distance connections within the brain. So this has become a daunting task for the neuroscientists to actually make sense of this connection. Who is talking with whom, and how often they talk, and what happened to these talks during the brain development when we age and things like that. So it's kind of one of the fundamental questions neuroscientists are asking. The neurodegenerative disorders such as Alzheimer's disease, these kind of disorders alter the brain structure. For example, in the case of Alzheimer's disease, there's a major reduction in the volume of the brain. This kind of a section of the brain showing what happens with Alzheimer's disease. There's a major reduction in the overall brain structure. The brain structure, the specific structure of the brain is because of the neurons and non-neuronal cells being organized in a certain manner. Decades of research into the mechanisms of memory has identified 2 major mechanisms. For example, one, they involve remodeling of these connections and making new connections. These are much like, when we have a guest coming, we try to rearrange our one of our rooms to accommodate something, or we wanted to build an extra couple of rooms to accommodate some permanent guests or something like that. So these things like this indeed happens in our brain. So there is a structural change in the brain. To symbolize this thing in a cartoonish way, in a highly reductionist way, this is one of these neurons called a signal sending neuron. This is a signal receiving neuron that will eventually go through kind of a structural change. So what is fascinating about these connections is that when we learn something new, there are changes in the cell body for producing new RNAs and proteins, and find that eventually leads to a changes in the connection with this target neuron. So my lab is trying to understand, how these connections are made and what kind of materials are being sent from the soma of the neuron to the synaptic terminals. Basically, if you want to be building something new, you need to have the materials. And these materials can be either produced locally or shall be shipped from far away from the soma. This is not just the story, but this is indeed really happens in our brain. I just want to give you an example of this. This is a kind of a traffic in the LA where you will find the northward and the southward traffic, they behave in a very nice way. And this kind of traffic indeed happens in the neuron. Here this is called a single sensory neuron of a aplasia, where you will see these membrane-bound vesicles are being traveling back and forth along these highways that are not really visible in these neurons. Over here, this is a single type of this vesicle is being shipped back and forth from the soma to the terminals. You can see that traveling back and forth. So these kind of things are happening in each of our 80 billion neurons. You can imagine that. That's why the neuron is highly microscopic, but it's a very busy place. It's full of activities in a very, very small space. To understand this kind of transport in setting up memories, so my lab used different animal models. For example, we use sea slug because of the gigantic neurons, and mouse because this is a mammalian model for learning and memory. What found is that this kind of a transport is mediated by specific proteins called kinesins. This is called kinesins. We found that these kinesins are important for setting up these connections in both components of the circuitry. And what's so fascinating is that we found that making materials for making the synapses are being shipped from the soma to the cell, to the synaptic terminals, including so many large number of RNAs. We found that this principle, this discovery, also holds true in the case of mouse. That is, a particular small protein called kinesin that transport a number of materials for making a functional synapse. So these are different RNAs that are being transported. So this will get to, this is leading to a major question like what RNAs are being transported. And RNAs could be like coding RNAs such as the messenger RNAs and non-coding RNAs. There are different classes of non-coding RNAs. We all know that the messenger RNAs code for proteins which are being used to build up cells and enzymes and things like that. The topic of today is about a particular class of non-coding RNA called the link RNA, the non-coding RNA. But these non-coding RNAs could be basically grouped into the housekeeping or regulatory non-coding RNAs. These ribosomal RNAs or the transfer RNAs, they are involved in translation, whereas these regulatory RNAs are involved in a number of processes, including post-transcriptional regulation, RNA modifications, splicing, and things like that. And the link RNA is the focus of today's talk, and I will be talking here through a much more detailed view of the species of RNAs called the non-coding RNAs or the link RNAs. They are defined arbitrarily as RNA molecules that are more than 200 nucleotides in length. So studies using different systems, particularly from the cancer biology and the developmental biology, they have discovered a number of functions of these linked RNAs. For example, they are in the nucleus, they function for transcriptional modulation, chromatin in the epigenetic modulation, RNA splicing, the cytoplasm, it is involved in regulating translation, the stability of mRNAs, and also function as sponges of microRNAs. And today's the talk, I will take you through three different aspects. First, I will take you through the link RNAs and the regulation of transcription in neurons, followed by the link RNAs and the long-term memory storage, followed by a brief summary. We were looking for long non-coding RNAs that may be important for synapse function. A small emphasis that synapses is the target of regulation for long memory. So a postdoctoral fellow in the lab, Ravindra, she has identified a particular link RNA called GM12371, and which we found that whose expression is regulated in the presence of cyclic AMP. And if you inhibit this particular link RNA by knocking down, that is reducing the level of this particular RNA, it impacts the neuronal arborization and the number of synapses it's going to make. Furthermore, it also affects the synaptic transmission between these different kinds of neurons. So here is an example like a link RNA that is being produced by a learning-dependent mechanism that actually modulates synaptic connections and looking at overall neuronal morphology. And it also not only impact the neuronal morphology, but also impact the communication between these neurons. Then we looked at what mechanisms are underlying its role in the structural change and in the synaptic transmission. She found that this particular link RNA born on chromosome 4 that regulates the expression of number of the mRNAs and other long non-coding RNAs. For example, it regulates many RNAs involved in cell function maintenance as well as this nervous system development and function. Here we have identified a link RNA that presumably that is enriched in the nucleus. That's why it's in the transcriptional control and regulating many of the genes involved in synapse function. Just want to take you through this thing a little bit more, this observation, a little bit more in detail. In response to cyclic AMP signaling, PK gets activated, that leads to enhanced expression of the link RNA from chromosome 4 that regulate the expression of two other link RNAs called GM10863 and GM13292 in a trans mechanism manner. So the name of these different link RNAs are not important right now. We just want to have this idea that link RNAs expression changes in response to learning, and the link RNA can regulate genes born on different chromosomes and affecting their function. This means it is a complex network of things is regulated by just by an expression of a link RNA. And we have studied this by very detailed techniques such as chromatic immunoprecipitation experiments, where you find that this particular promoter for this particular link RNA become enriched during activation and RNA become enriched upon activation, suggesting that this particular link RNA is activating RNA expression of other link RNAs and mRNAs in the nucleus. So this is a very good example of a transcriptional control of synapse function, that is transcriptional control of neuronal connections and neuronal communication by a link RNA. Now come to the part 2. This is about the specific functions of link RNAs and learning. Dr. Espadas in the lab, who worked on this project, what she did was she carried out a behavioral training experiment. This is called the contextual fear conditioning. So this involves basically you place a mouse in a cage and you give an electric shock, and the mouse learns that the cage is an unfriendly environment. And when you put back the mouse in the particular cage, it remembers it's shown by the freezing of his body. This is called contextual fear conditioning. And we all know that we all experience some of this behavior, this kind of learning in our own lives. For example, if you have a bad experience in a particular room, when you go back, that kind of try to keep on haunting you, right? So something like that happens in mouse. And we can actually, and the memories of that fear in mouse can actually last for months. So to understand the role of these link RNAs in learning, Dr. Espadas did this kind of a behavioral training. And after that, she isolated a small region of hippocampus. That's the place where the memories are stored initially, by using a technique called the laser capture microdissection. This involves using a laser. You isolate a very small area. And that's what you see here. This is after dissection. Then you sequence these using these next generation techniques. And she has identified a number of genes, especially the link RNAs, that become upregulated or downregulated in response to this kind of learning. And one of this particular link RNA is called the D17. And that's what she tried to study the function of this link RNA D17 by implanting a cannula, and that will help for us to inject this particular antisense oligonucleotides into the mouse brain, and you can test the functionality of the link RNA in behavior. And what she did was she looked at multiple behaviors such as acquisition, consolidation, and extinction recall. These are different behavioral assays. For example, when we try to learn something new, there's an initial period where things need to settle down. That's called acquisition. And after that, repeated trials of this particular training would help us remember. And how much you will remember, that we need to be testing after a certain time. For example, a day later, that's called consolidation. And you can say that you can actually try to learn something completely new. For example, that bad experience was not really a bad experience, something like that. That's called extinction. And these are different kinds of behavioral testing to see the role of this particular link RNAs in memory. But these behavioral paradigms are nothing new. These are actually kind of a standard behavioral assays. So we were using the standard behavioral assays to study the function of a long non-coding RNA. And we identified in a specific subregion of the brain called the CA1. What she found is that when you inhibit this particular link RNA, there is no change in the acquisition or extinction recall, but only for the consolidation of memory, suggesting it has got a major role in setting up, in consolidating this long-term memory. So here we go. Here we have one particular link RNA that is expressed in a specific subregion of mouse brain, and it is critical for setting up memory. So we want to know how this link RNA, what makes this link RNA unique? How this link RNA is able to mediate all these, its role in memory process. So what we did that was to actually isolate these complexes, anything that is talking to this particular link RNA. We identify them and we tease apart. For example, all the RNAs that are interacting with D17 were identified by total RNA-seq. This will help to identify other microRNAs and mRNAs and link RNAs interacting with D17. And we did mass spectrometry to identify many of these proteins. So these kind of very in-depth analysis resulted in identifying about 33 different link RNAs and 10 microRNAs being enriched with D17. So I think it's a very intriguing complex. Over here, it's a particular link RNAs interacting with dozens of other link RNAs and other dozen of microRNA and several other proteins. So this is actually giving it a kind of almost a nightmare. So we still don't know like how this is able to exert its role in memory. So we decided to do one experiment. So that was what if we isolate these link RNA protein complexes and we try to see whether what regions of the link RNAs are interacting, whether that may be important, that can give us some clue. So that is indeed gave us some clue. We identified two different regions in this particular link RNA that is interacting with protein. For example, we found to be impacting CAM kinase 2 and vimentin. So these proteins are very important for synaptic signaling and structural plasticity. So here we tend to get the mechanisms by which there's a dedicated regions within this particular link RNA interacting with proteins, and that interaction is kind of real based on these experiments. These experiments also tell us that this protein may be this particular link RNA D17 may be expressed in neuronal, in dendrites, may not be in the nucleus, and we proved that by a number of experiments. So one of the experiments I want to take you through is called visualizing of this transport. This was done in collaboration with another postdoc in the lab, Dr. Wickfield. So they used a technique called MS2 based tagging. Over here is this particular link RNA that's being tagged with phage sequences, so such that GFP, this MS2, this complex actually formed at these UTRs of this D17, and when this link RNA moves in the cells, because of the GFP, we will be able to track its transport. And this is indeed, we actually could make that happen. We could actually see that the transport of these link RNA particles in hippocampal neurons. You can see that these white particles, each one of them is is a link RNA particle coming. We can see that coming from the cell body to the processes, and from the processes to the cell body. Over here, it could be a little bit clearer over here. You can see these molecules are being long distantly traveled towards the terminals and also to the back. So we show that the link RNA produced from the cell body is being shipped down as RNA to the synaptic terminals. We have done a number of experiments to prove that this transport is essential actually for making the synaptic connections. And if you remove these 200 base that's interacting to CAM kinase, if you remove that, you actually end up blocking this particular transport. And one of the fascinating thing about this particular transport is that when we imaged, we found that this transport is more towards this place where there are connections between neurons. The PSE95 is a protein, it's called postsynaptic density protein 95. This is enriched in these synaptic connections. We found that by quantitative imaging at the D17 RNA become more localized to these synaptic terminals. So this suggests that it has a function at the synapse. And we wanted to know whether this is actually critical for setting up these connections. I mean, highly likely that it is important. And this is exactly what we found. We used different kind of constructs. So this is a knockdown construct to reduce the levels of this particular link RNA. Over here is an inducible knockdown. Important thing is that if you knock down this particular link RNA, it affects the arborization, the number of branches these neurons can make, and the synaptic connections. Then we carried out a little bit more sophisticated experiments, that is by just stimulating one spine, one synapse, and see what would happen when you knock down this particular link RNA. What we found is that when you knock down this particular link RNA, the synapse enlargement is impacted for the fast change in the synapse structure as well as the slow change in the synapse structure. So there is a huge change in this connection with this D17 modification. So that's why we conclude that this particular link RNA is critical for making these connections and modifying these connections. To summarize, I have shown you some examples where this link RNA expression could be cell-specific. It may be present in the nucleus versus cytoplasm. It can be temporarily regulated. Its interacting partners could be very complex. It can involve microRNAs, link RNAs, mRNAs, and proteins. And there are dedicated regions within each of these link RNAs that mediate this interaction. It may have this function at the cellular and systems level. This is just a tiny bit of an iceberg. This is a tip of an iceberg. There are a lot more to study, so we are actively pursuing these studies and using C-SLEX and mind. And right now we are also trying to study these things, mini brains, human mini brains. With this, I want to say that, I'm very, very fortunate to have wonderful people working with me, and I have mentioned the name of the scientists who were directly involved in this particular study. I shared with you, and I'm also blessed with a number of funding from the federal agencies. And as you can see, that conferences, something such as this, this need to bring together like-minded people to make something bigger and better. And this is what I have been trying all my career so far. I've been fortunate to have all these fantastic collaborators. I just want to say thank you. And again, I'm going to say that I'm sorry I'm not there in person. I wish the very best and exciting meeting. Thank you. Bye.
Thank you,
My
Dr. Satya,
apologies.
for
Sorry, my apologies. Wherever you are, these kind of technical issues can happen any corner of the world. I mean, I'm very sorry that I really wanted to present in person. I mean, it couldn't happen, but I'm glad that the video kind of worked out. Thank you for tolerating.
Thank you, sir. It was a very enlightening speech, and we greatly appreciate for spending your time with us. It's midnight over there, and still you are with us. It's really appreciated. And with increasing cases of Alzheimer's and Parkinson's around the world, the work of neuroscientists becomes indeed very important. And thank you for making us aware of the work of the link RNA D17, sir. Thank you very much. I hand over the mic to Benazir, ma'am.
Even we thanks Dr. Daniel sir for his presence.
Thank you.
Thank you, sir.
Thank you. Now I request Professor Dr. Vijay Desle, sir, to introduce our invited speaker of today's session, Professor Suvarna Kulkarni, Madam, from IIT Bombay. Over to you, sir.
Hello, good morning all of you. It is my great privilege to introduce Professor Suvarna Kulkarni, sir. Sir has completed his BSc and MSc from University of Pune. Sir also completed 3 from University of Pune. Then sir joined for postdoctoral research in Academia Sinica Taipei under Professor Shang Hung from 2001 to 2005. Then sir worked as an assistant specialist at University of California, Davis under Professor Haig from 2005 to 2008. Then he joined as an assistant professor in Indian Association for Cultivation of Science from November 2008 to 2009. Then sir has joined IIT Bombay in February 2009 as assistant professor, and currently he is professor in the Department of Chemistry at IIT Bombay since 2016. Sir is also Associate Dean of Academic Program and Institute Chair Professor at IIT Bombay. Sir is a life member of CRSI, Association of Carbohydrate Chemistry. Sir is a member of American Chemical Society, Fellow of Royal Society of Chemistry, and Vice President of Royal Society of Chemistry, West India Section. Sir is editorial board of member of Carbohydrate Research. It is elsewhere journal. Sir has received numerous awards. To name few, Professor Sukhatme Award for Excellence in Teaching in 2020. Sir has received CRSI Bronze Medal 2017. Sir has received Dr. H.C. Srivastava Young Scientist Award in 2016. Career Development Award by University of California. Young Faculty Award by IIT Bombay. He has published more than 80 research publications in reputed international journals and more than 2200 citations. Under his guidance, 9 students have completed PhD. Currently in his research lab, 17 students are pursuing PhD and 2 students are doing postdoctoral research. Now I request Professor Suvarna Kulkarni sir to deliver your talk. Over to you, sir.
Thank you, Professor Vijay Desai, for a very kind introduction. At the very outset, I would like to thank the organizers, especially Dr. Kirti Patel, who is our alumnus, and Dr. Kulkarni madam, for giving me this opportunity to visit your college in a virtual mode to share some of our research. And it was really nice to see Dr. Satya's work, a very cutting-edge work about related to the brain science. So today I'm going to talk about sugars. And my topic is, are there sugars in bacteria? What can we do with them? So this is the work that we have been pursuing at IIT Bombay over the last 10 years. And this is the overall theme of our research. As all of you know, these infectious diseases are on rise and they are constant threat to human beings. And antibiotic resistance is also the rise. So one question is that can we develop novel antibiotics, therapeutics and vaccines from carbohydrates? And can we do this by using chemical synthesis? So before I begin, just wanted to make sure that I'm audible and the slides are visible.
So you are audible and the slide also visible.
Thank you.
Thank you. Continue.
Thank you. So how we can involve chemical synthesis, because that is our expertise. So this is a view of our cell with the nucleus, and these are carbohydrates. And as you can see, these carbohydrates are ubiquitously distributed on the cell surface, where they can participate in a variety of vital life processes, such as viral and bacterial infection, cell-cell communication, immune response, to name a few. And structurally, they are diverse and complex, and they are present in microheterogeneous form in nature. This is another cartoon picture showing you the placement of carbohydrates, where carbohydrates exist as glycoproteins, where sugars are attached to proteins, or glycolipids, where they are directly attached to lipids. And you can see that they are perfectly placed in such a way that they can play key roles in anything that has to do with the cell-cell interface. Right? So it was believed until 2000 that these sugars, which are the post-translational modification of proteins, are only present in eukaryotes, like in us, and they're virtually absent in bacteria. But in the last 15-20 years, this notion has been refuted, and it's now well established that the sugars are also present in bacteria and even archaea. So this is a TEM picture taken from Israel in 2006, which shows that you can see this is a bacterial cell, and these villi are nothing but the glycans which are protruding out of bacteria. So now it is very easy to understand that when our cell, our immune system encounters this bacteria, the sugars is what we see, what our cells must be seeing at the very outset. So this gives them very crucial place in drug discovery or drug development. Now there's a difference though. While mammalian cells, they have a very few kind of standard sugars such as the glucose, glucuronic acid, galactose, and so on nanos. Bacteria often have hundreds and thousands of variety of sugars with various modifications which are virtually absent in human beings. For example, these 5 sugars I have written here. So this is the standard sugar GalNAc, and bacteria sugar often have some of the hydroxyl groups modified as amino groups, and some hydroxyl are removed like a deoxy sugar. So these are like AT, DAT, DG. These are the short forms of fucosamine or bacillоsamine. These are the sugars which are present in select group of pathogenic bacteria. To give you a few examples, for example, this DATDA sugar is present in Neisseria meningitis species, and bacillоsamine is present in Campylobacter jejuni. This is a glycan of Campylobacter jejuni. So this is the protein of the bacteria, and the sugars are hanging down. And our eukaryotic glycans, they have this N-acetyl galactosamine. Also, this Pseudomonas aeruginosa, it has a D-fucosamine as a sugar. What is more important is that the presence or absence of these sugars has been shown to be linked to pathogenesis. Okay. But the problem is, so these, because bacteria have these sugars and we don't have these sugars, they are very good candidates for vaccine development. However, in order to make a vaccine out of carbohydrates, they have low antigenicity, and we need to tag them to carrier proteins such as tetanus toxoid. And we also, our immune system also reacts to that protein. So we also get antibodies for that protein. So to counter this, Dennis Casper and others from Harvard, they found out that oligosaccharides which contain charges like deuterionic oligosaccharides, you don't need to tag them to carrier protein. They can directly bind to T cells and invoke a strong immune response. And these deuterionic oligosaccharides obviously will have these rare sugars present in them. So to summarize this slide, I would say that these sugars are not present in human beings, and they are present only in bacteria. And so they are very good candidates for target-specific drug delivery and vaccine development. And so what we thought is, what is required is we need to get access to this pure compound, because we cannot get it by isolation from bacteria as you get a micro-heterogeneous mixture. So in order to do biological activity, you need pure compounds. And we thought that we can make these compounds, but for that we need this access to blue-colored galactosamine building blocks, which are orthogonally protected by variety of groups. And we also need access to these amino sugars, deoxyamino sugars. So we began our research in 2011, 10 years ago, with these sugars. And we asked, first thing we do is literature survey, and we were very surprised to see that there is only handful of exhaustive list of references, not even 10 references from all the leading labs in the world. And these methods describe synthesis of these sugars starting from carbohydrate precursors. And then Peter Seeberger and others started publishing around that time the de novo synthesis, like they will start from amino acid or L-threonine or garner-aldehyde and then build this sugar. So these are also very long processes, and it gives you a mixture of distinct. So what occurred to us at that time, that the general route to all the rare sugars and glycosamine building blocks is missing. So this was our idea. We thought that, okay, we can begin with a simple sugar D mannose. We can carry out, this is the primary hydroxyl groups, and these are the secondary. So we can do selective deoxygenation here. And then if we could find a method to selectively mask this hydroxyl group by taking the help of syn disposition of the diol, we'll be able to get this 2,4-diol, which we can then convert into good living groups such as triplet. And then if we can carry out selective SN2 displacement of one of the triplets, and then the second triplet or good living group, we can then do one-pot reactions. And by changing the nucleophiles, we can get access to all the rare sugars. So this was our plan. As I mentioned, the first reaction worked really well. You do selective tosylation and reduction with LH to give you the deoxy sugar. The next reaction, it took us some time, but we figured out by using Onomura's conditions and our modification, we could mask this C3 hydroxyl group either as acetate or benzoate. And the reaction really works well. On a gram scale, you can get 0.8 gram of diol. And this reaction takes place because of the presence of catalytic amount of dimethyl tin dichloride, where the tin coordinates this syn diol, and you can get the selectivity at this position. So at this position, at this time, the stage was set for nucleophilic displacement. And as you can see, if you imagine the SN2 displacement, we thought that for the top phase attack, there is a steric hindrance of the C3 triplet. But from the bottom phase attack, there is no steric or electronic repulsion. So the first reaction would take place easily, and then we can add a next nucleophile in the same form. But the problem is, as soon as the first leaving group departs, the intermediate competes with the substrate, and you may get a di-displaced form. But nevertheless, we thought that by playing with solvents, we'll be able to solvent and reaction temperature, we'll be able to obtain the selectivity. So this was our idea. As you can imagine now, if you imagine a nucleophile coming from the top phase, right? It will be hindered by the C2 triplet electronically as well as sterically. So from a bottom phase attack, there is no repulsion here, and it will give you displacement of this triplet. And once you get this, now the next nucleophile, let's say a nitride, can come from top phase, displacing this, giving you the desired di-displaced or double inverted form. So this was our idea, and yes, indeed, Madhu Emadi, my first student, he worked on this project, and he struggled for 6 months, and he found out that if you use, when he started using sodium azide, he was getting a di-displaced product to 20% extent, along with the desired form. So he just used excess, and we got this diazodo product, which is also important. But after struggling for some time, Madhu was able to show that if you use a bulky azide, tetrabutyl ammonium azide, we can then displace selectively only this triplet. And then he could add another nucleophile in the same form. So what you're doing is, in a one-pot manner, you are inverting these two centers and getting nitrogen differentiated functionality in one-pot manner. So this was a remarkable result which we got at that point of time. It was also appreciated well by the scientific community because that time there were no methods for this. And then you can change the second nucleophile to tetrabutyl ammonium nitride, and you can get OH over here, and you can do one more iteration of reflection inversion to bring azide equatorial fashion here to get a bacillozamine. Well, if you want a fucosamine with a protecting group at this position and not this position, what we do is we add water as a nucleophile in excess and heat up the reaction. This reaction takes place via orthoester formation, and the orthoester opens on the axial phase, giving them this base. So all this works really well. And how well this works, this di-displacement reaction, to give you an idea, from a diol to rare sugar building block on a gram scale, you can go in 1 to 2 days time and in really excellent yield, and you just have to do one column at the end of it to get the pure compound. So at that, when we also asked that this is fine, we are getting these rare sugars, can we also get the galactosamine building blocks using the same technology or methodology? And the answer was yes. For this, we don't do deoxygenation, we do TBD-based protection and repeat everything just the same way to get us access to all these orthogonally protected galactosamine building blocks. So at that point of time, in 2013, what we had in hand is a very elegant access to start from a simple D-managed site and get access to all these variety of rare sugar and glycosamine building blocks as a thioglycoside donor or acceptor, which we can then use in total synthesis or metabolic labeling experiments for selective detection and selective disarming of bacteria. Now I'll speak to you about photosynthesis part first. We have accomplished some 20 total synthesis in last 3, 4, or 5, 6 years. And I'll first show you just the structures because of and then I'll show you some one or two representative examples of photosynthesis. So this trisaccharide was completed by Madhu Emadi in 2013, and you can say it has both sugars, the bacillozamine as well as BA2BH. And this, which has a pseudo trisaccharide, is completed by Anandrav Podilapur, and this more recently by Palasaheb Gotekar, which has this AAT as a sugar building block, and this is from Streptococcus pneumoniae and Pyoralis. And I talked to you about these deuterionic molecules Our lab is one of the first labs actually to complete all of the photosynthesis of all the molecules. So you can see, and these bacteria, they really have very, very rare appendages. So you can see it has AT as well as you have a phosphoglyceramide linkage, you have carboxylic acids, and this gives them unique immunological properties. So that's why we are interested in it. And most of the compounds, you can see here, you also have a phosphoethanolamine, or here you have a furanoside and the glucuronide, galactoronide. So, but here in this case, you have a pyruvate. So, these total synthesis were recently completed, and we have also synthesized some more exotic glycans where you have rare amino acids, phosphates, and so on. And Madhu MRD, just before giving finishing touches to his PhD thesis and joining Professor Professor later, he also completed the hexasaccharide synthesis, which has more rare sugar. So at that point of time, we started wondering, are there L counterparts of the sugars in bacteria? And we were really happy to see that bacteria also has L glucosamine, L fructosamine, L pinosamine, and so on. And there are no methods to make these compounds. So to the second student, Somesh Rao Sanapala, who is now faculty at IIT Tirupati, I asked this question: can we use this chemistry and we can start with L-rhamnose, and then we can make all these real sugars by using the same method. You protect this, then you do triflation, and then you do inversion, right? And if this works, then we will use isomer of L-fructose, isomer of L-rhamnose, where the 2 and 4 positions are interchanged, and then do the same chemistry. The idea is that each time the actual triplet will undergo displacement first. And then we can have the flexibility of changing the nucleophiles to get access to all the deoxyamino sugars. And to cut the long story short, Someshwar Rao Sanapala was able, single-handedly he was able to convert these two sugars into all these rare and sugar compounds. So then, having these both L and D sugars in hand, we started wondering, are there glycans wherein you have a mixed D and L sugars? And if it is so, then we can use it for testing our methodology to construct these glycans. Now, around that time in 2017, WHO came up with a list of priority pathogens, and WHO urged, World Health Organization urged all the scientists, scientific fraternity, and industry people to work on these multi-drug resistant pathogens such as Pseudomonas aeruginosa, Staphylococcus aureus, and all the grouped under escape pathogens. So this one, for example, Pseudomonas aeruginosa, it has this trisaccharide repeating unit where you have a D-fucosamine and L-fucosamine combined together in an alpha-stereoselective manner. So we thought that we can make this, and we should also add one amino propyl linker for conjugation with a protein, so that we can test the immunological properties of these compounds. So this is listed as a critical priority pathogen by WHO. Similarly, Staphylococcus aureus type 5 and 8, they have also been listed as a MDR high priority pathogens. And it also has the same disaccharide, but the third sugar is different. It has a manuronic acid, manuronic amine acid, which is challenging sugars to make actually. And so just to give you an idea about the efforts that the students put in the lab, I would like to describe the photosynthesis of these two compounds, which has this common disaccharide in these few minutes. So again, in short, I have explained to you that how we convert D-mannose into D-fucosamine and L-rhamnose into L-fucosamine, and we use simple regioselective protection and SN2 displacement reactions to get to this. Now the key step in this synthesis was coupling of this thioglycoside, this carbon atom, with this hydroxyl in a stereoselective manner. Okay, and we could do that in 94% in only alpha and H1. So this was a very elegant way, and two students, Archana Behra and Diksha, are working on this project, discovered that we need a benzoate group which can exert a long-range electronic effect, stereoelectronic effect, to get alpha selectivity. And then once we get this disaccharide, we then elaborated them into these two glycans. And in the next 2-3 slides, I will explain the total synthesis for those who like. Now, this is what I was talking about, the stereoelectronic effect. So you have a benzoate here, Once you activate this thioglycoside, so lone pair on sulfur will attack the I plus, and it will be dislodged by this lone pair of oxygen to form oxonium ion or oxocarbenium ion in this case, which has a flat structure. It has a half chair structure. So if you imagine this, the equatorial rich structure will be predominant, and either way, axial rich or equatorial rich, you will have a steric effect and electronic effect from the bottom phase. So the nucleophile can only come from the top phase, because this benzoate group stands in the way of the nucleophile if it tries to come from the bottom phase. So you get, in this 3H4 conformer, you get top phase attack leading to this alpha glycoside. Now, this is a synthesis done by Archana. So what she did is she did essentially a regioselective glycosylation on this diol, where equatorial hydroxyl group is more reactive. So she got this as a major product in 73% yield. And then the benzoate was removed by sodium methoxide, and this hydroxyl group, then this N3 and NHTCA were converted into NH acetates under reductive conditions, and then hydrogenolysis removed the remaining functionalities to give the final molecule, which we then handed over to our collaborators for computation with proteins and for immunological studies, to Peter Seeberger at Mehta. So this was more challenging. The Staphylococcus aureus type 5, this was done by Diksha Rai. From at that time she was just in the second year. So the way it happened is, in her case she wanted to couple the less reactive axial hydroxyl group, so she cannot do regioselective glycosylation. And here, how in carbohydrates the distant groups play a role. So this, when she had this acetate in the neighboring group, she got the trisaccharide, but in 40% yield. Now this is a disarming group because it pulls the electron density away from this nucleophilic alcohol. So the moment she changed this acetate into the naphthyl methyl group, the naphthyl methyl group gave arming effect, and she was able to get excellent yield, 84% yield of this. Then here the benzoate group was removed, and then triflation and inversion on trisaccharide gave her this azide in the right position. The naphthyl methyl group was oxidatively removed by using DPQ and was replaced by acetate, because you have acetate in the final molecule. Now you remove the benzidine and do oxidation at this position, 6th position, to get carboxylate or ester here, and then you do a reduction of azide and NHTCA into acetates, and then you do global deprotection to give us this trisaccharide. So this was, again, I must say, that heroic synthesis from Archana and Diksha. And more recently, Balasaheb Gotekar and Shayantan Biswas, these two students, brave students, they accomplished the protein synthesis of this pentasaccharide, very complex one. Due to the time limitation, I won't describe the protein synthesis. If you are interested, you can pick up this paper. It took us 5 years to complete the protein synthesis. So at this stage, I would like to, maybe I can just show you briefly. So here you can see this bacillus-amine building block I've already told you, but here we need to differentiate both NHTC and N3 at this position, and this we can get by selective reduction of beta-keto ester, and this one we can get from mannose. These building blocks I've already told you how to make it by using double displacement. The problem here though was this, as you can see, 3 groups are axial and 2 are equatorial. So I was skeptical that maybe this sugar is not in this conformation, and it's in a flipped conformation. This is shown as 1C4, but I was thinking it's a 4C1, because in that conformation, these 3 groups will flip and will become equatorial. But Shayantan proved me wrong, and he got the X-ray crystal to prove that indeed this sugar presents in this conformation. And I will show you the X-ray crystal, which clearly revealed that this is the sugar molecule here. And you can see the azide is also down, acetate is also down, and here your PMP group is also axial. So it's an axial rich conformation, which was proved by the X-ray crystal above. Then rest, they did a coupling here, and again you need the anchimeric assistance, long range participating group. Then you remove this and again couple. So deprotection coupling gives us a trisaccharide, and then you add one more sugar to get a tetrasaccharide, and then pneumosamine building block to get a pentasaccharide, and video selective, and then global deproduction gives us pentasaccharide. So here I would like to change gears, and in next 15 minutes or so I will describe the metabolic oligosaccharide engineering. Now, what is metabolic oligosaccharide engineering? So because after doing all this total synthesis, we started wondering, these monosaccharides on their own can be used for detection of bacteria, because only certain bacteria have these sugars and we don't have them. So we resorted to this technique called as metabolic oligosaccharide engineering in collaboration with Daniel Dubé from United States. So this technique was introduced by Caroline Bertozzi way back in 2003 and Rauter. So the idea is that this is a bacteria and this is a sugar present on the bacteria. The idea is that you grow bacteria in the presence of sugar which has azide group on them. Then the bacteria will take up the sugars and use its biochemical machinery and incorporate our sugar in their glycan and then express it on the surface. Once this azide group is expressed on the surface, we can then intercept it with a bioorthogonal probe such as phosphine containing fluorophore, and this will lead into fluorescence, so we will be able to image the cell surface. So Bertozzi did this in mammalian cell. And my collaborator Daniel Dubé, she started working in H. pylori, which is a very critical bacteria, and detection of H. pylori is very, very difficult because it is you have to do endoscopy and other things to find out this bacteria. Stool examination is not that great. So there's a great interest in world to find out better detection tools for this H. pylori. So the idea is this. So she started with glucosamine and did a metabolic oligosaccharide engineering, and this was her method. So she would use a flag peptide, right, and will do Staudinger ligation of this phosphine with this azide, which is expressed on the bacteria, and then use a anti-FITC antibody, anti-flag antibody to do the imaging. So with this, Daniel Dubé discovered that this is a cool method. If you use glucosamine, you can facilitate detection, identification, and analysis of proteins in this bacteria, and very easy to detect this bacteria using glucosamine. So at this stage, we asked her, can we use this same sugar for reaching out to, how about the selectivity, whether this sugar goes in Pseudomonas aeruginosa, B. thailandensis, B. fragilis, and Mycobacterium spontatis, and C. jesuni, all the bacteria are more in equal importance. Then this was the idea. So grow each bacteria in the presence of only this glucosamine sugar with the azide handle. We were surprised, very, very surprised, and we learned a lot from this experiment. This is a Western blot experiment that is showing the expression of protein on the gel, and you can see only expression was seen on H. pylori, but no expression was seen on Campylobacter jesuni, Pseudomonas aeruginosa, B. thailandensis, or Mycobacterium smegmatis, telling us that glucosamine is taken up only by H. pylori and not by other bacteria. So the next question is, okay, so how do you find other bacteria? We have a method to find H. pylori. What do you do with the other bacteria? So for this, the simple solution was that, okay, we said we will look at what kind of sugars those bacteria have. If the bacteria have that kind of sugar, that bacteria must have the enzyme, so the glycosyl transferase, to transfer that sugar onto its protein. So then we made this, we synthesized a panel of these analogs, which are present only on those bacteria, and then we rerun our gel on this set of bacteria and our host cells. And now we are happy to see that these bacterial strains, all of them, they differentially incorporate the azido sugars. And depending on what kind of azido sugar substrate we use, it dictates which bacterial species will become azide covered. So this was a good result, and we checked whether these sugars are entering into our host cells. And this is a flow cytometry experiment which showed us this is a background signal, and there was no incorporation of these sugars in host cell. So now we found a selective method. Taken together, you can say that we found a selective method to reach out to bad bacteria and leave out the good ones. So when you take antibiotic, you kill all the bacteria in your flora. So with this method, we are able to do selective labeling of pathogens. And we believe that why we are getting selectivity is because each bacteria has a different set of glycosyl transferases. So using their own machinery, we are able to reach out to them and put a cap on their head so that even if they are in the crowd, we can find out. Now the next question is that now that we have identified these bacteria in a mixed population, can we selectively disarm these bacteria? Right? So in next few minutes, I will just wind up showing you the results of the selective targeting. So the idea is this. So if you have a bacteria, the bacteria has a fully decorated glycan on their surface. And this decoration is essential for for bacteria to cause disease. So we thought that if we can make an inhibitor based on the same rare sugar, their own sugar, and if we can mess up or interfere with their glycan assembly and lead to the truncation of glycan, this will have impeded fitness and this will not be able to cause disease. Okay. This is a typical idea of any antibiotic development. Drugs like vancomycin work on this principle. So our idea was to use these benzyl analogs. So where bacteria will take up as a substrate and do glycosylation on our substrate, and therefore its own protein will remain devoid of sugars and will have truncation or less expression. The other idea is that you use it as a chain termination. So this will go on bacterial protein, but then this fluorine will stop the further glycosylation. So here I'll show you a better picture. So if you have a benzyl, so basically we are diverting glycan assembly away from the bacterial glycoprotein in this idea. And we use these acetates because they're useful for passive transport across the membrane, and you have endogenous esterases which will remove these acetates. So this was one idea. The second idea is chain termination. So this would be accepted as a substrate, will go on the protein of bacteria, but then bacteria will not be able to decorate it further because the OH is replaced by fluoride. So this was our idea, and we thought that we can check this by using the diminishment in the MOE based signal. So without our rare sugar, we will see metabolic labeling, we will see incorporation, and then we will use this benzyl group, benzyl glycoside and fluoroglycoside together with the rare sugar. And if there is a diminishment, we will know that this sugar percentage is decreased. And this is one result I want to show you. You can see these benzyl bacillozamine and benzyl fuconate. These two showed dose-dependent diminishment in the concentration. Complete diminishment, right? And complete diminishment also in case of fluoride we got good results. And then we did lectin binding assays which are not shown here to confirm that indeed bacterial surface has been changed and the glycosylation pattern has been decreased or changed. So now we want to know, it's fine, we confirm that using this technique we are able to mess up with the glycan assembly and lead to truncation of glycan on the surface. So what is the use? Is there any effect on growth, biofilm formation and motility? So for that we did these three experiments. We did optical density 600, then crystal violet stain and halon agar sugar. And you can see here this is a growth curve. This is a standard wild type growth curve, right? And this is with our inhibitors. You can see completely growth halt with FDAT and benzyl back, right? And this is the biofilm, complete clearance of biofilm. And also you can see here this FDAT and benzyl back outperforms even the mutant of glycosyl transferase. So this is very, very good. And we could also see in case of benzyl fuconate, also similar results were obtained. So you can see the biofilm were quite clear. But taken together, we realize that in case of H. pylori, these inhibitors can inhibit the glycan biosynthesis, and that leads to growth, biofilm formation, and motility defects. Well, the same effect was not observed in C. jejuni, and in B. fragilis, we observed some subtle differences in glycan biosynthesis. So what we proved through this study you can take it as a proof of concept study that you can really alter the bacterial glycan surface, and that will lead to the failure of that bacteria in causing infection. I think in our opinion, this is the first example of using metabolic oligosaccharide engineering and unnatural monosaccharides to inhibit bacterial glycan. And this is a strategy to harness bacterial glycan to selectively disarm bacteria. We are not claiming that we are killing the bacteria, we just stop their growth. And we believe that these inhibitors, if you take it as if you co-administer them with antibiotic, and they are not toxic. We have studied the toxicity profile. These are not toxic. So you can take them together with antibiotic, and these inhibitors will make the bacteria more vulnerable to antibiotics and also to our immune system attack. Where we go from here? So we are going towards development of narrow spectrum antibiotic against high priority pathogens. And why this method is so cool? Because you don't need to have any information about the glyphosate transferase pathways. It's just like fishing out. You really need to know what kind of fish are there. So now we are going ahead with this bunch of important bacteria, and further we are also developing fluorescent sugars so as to find out exactly what is the fate of these sugars inside the bacteria. Well, this study has received a lot of attention from international and national community, and we are very pleased about this. So there was an article in Chemistry World that said that sugars designed to destroy bacterial cell wall biosynthesis. Then this story was picked up by another international journal, Research Matters, and then more recently, Hindu Business Line, they started a science topic in their newspaper, and then ours was the first one to display bacteria killing sugars. And then of late, it also came in the local newspaper, like the Sakar newspaper, the front page, and this is in Lokmat and other newspapers. So all this work, none of the work has been done by me, right? I'm just like a spokesperson of this party. I'm privileged to work with. So the work was started with Madhu MRD, who did all the work on D sugars, and then followed up by Someshwar Sanpala, who worked on the L sugars, and then Anidrav Todilapur, these three students, who worked on a variety of glycans actually. And this is Archana Bhera, who did the synthesis of the trisaccharide. Now, where are these people now? So Madhu Amadi went on to work with Seeberger, and now he's working in vaccine company in Germany. Someshwar again went to Seeberger, but came back recently and joined a faculty position last year in IIT Tirupati. Anandrav Podilapu is working in Georgia as a postdoctoral fellow with MG Fin. Archana Maibera right away got a assistant professor position in Utkal University, so she did not go for a postdoc. And this is my more recent group. Diksha Rai, I mentioned, Ankita Paul, who worked on these real sugars. And so with this, I would like to once again thank all my students and all the funding agencies, and most importantly, Daniel Dubey for her collaboration. And once again, thank to all of you for your patient listening and for giving me the opportunity to share our work. Thank you.
Thank you, sir. The speech was very much informative, definitely informative speech. As we all have gone through COVID pandemic, we are all aware of the importance of development of vaccines, drug development, and narrow spectrum antibiotics. Definitely, sir, your research work and your students' work is remarkable. Now I ask Dr. Desley, sir, to give thanks. Over to Dr. Desley, sir.
Thank you, madam. Sir, thank you very much. Your talk is very informative and interesting. You have enlightened us with a great knowledge of carbohydrate chemistry. Being a synthetic organic chemistry, I know how to understand the even simple structures of carbohydrates. But you have worked a lot on research of the carbohydrates, and you made this very simple to understand the stereochemistry as well as regioselectivity or regiochemistry of carbohydrates. As explained, recognition of carbohydrates on surface of the cell, He has also explained bacterial sugars, bacterial glycans, regioselective reactions. Sir explained very nicely one pot protocol for rare sugars and their efficient synthesis, total synthesis. Sir also delivered very nicely metabolic labeling experiments, deuterionic molecules, rare deoxyamino sugars, glycosylations, and their anchimeric assistance. Sir explained very nicely metabolic oligosaccharide engineering. Really, sir, lecture was very interesting and enlightened us and all the participants with a great knowledge of carbohydrates. On behalf of NB Mehta Science College, Bordi, and all the participants, I express my sincere gratitude to you, sir, for delivering very nice talk on carbohydrates. Really, thank you, sir. Thank you very much.
Thank you. Thank you.
Thank you, sir. Now, if any one of the participants has got any questions, they can write them in the chat box. I think sir can answer some of the questions. It was not possible with Professor Satya because it was almost 2 a.m. over there in Florida. But you can ask a few questions. If you have any, you write the questions in the chat box, please. I think it was so nicely explained by you, sir, that at present the participants are not having any questions, but I'm sure that they can always communicate with us and ask all the questions, and we can convey those questions to sir. Thank you again, Dr. Kulkarni. It was indeed very nice.
Thank you. It was wonderful to interact with all of you in a virtual mode, and I look forward to visiting your college sometime in future. Thank you.
Definitely, sir. Definitely. You're most welcome.
Thank you.
Now we move on to the oral presentations. As you all are being informed, there will be 3 sessions. Session A is of life science topics, including microbiology, zoology, and life science. This session will be chaired by Dr. Nitin Patil, and Ms. Bhumi Das will be the rapporteur for the session A. Session B, that includes biotechnology and botany, that will be chaired by Dr. Shilpa Bharat Madam, and Mr. Nikhil Morankar will be the rapporteur. Session C is of chemistry. The chairperson for this session is Professor Shivram Garje, and Ms. Benazir Sheikh will be the rapporteur for this. The time allotted for each presentation is 5 minutes, and maximum of 2 questions will be allowed. Session C, that is chemistry presentations, will continue on this same link. The links for the life sciences, that is session A and B, are already sent in the respective WhatsApp groups, and the same can be viewed in the chat box also. I wish all the participants a very good luck and hand over to the rapporteurs to conduct the sessions. Thank you all. Thank you.
Yeah. Benazir madam, I'm in. Can we start the session? Or we have to wait?
Hello, Benazir. Please answer his question. Benazir, when are we starting? Benazir,
Yes, ma'am.
can you hear me? Can you hear me?
Yes, yes. So, within a short while, we are going to start with the oral presentation session. All are requested the participants of session C are requested to remain in this group. Within a short while, we are going to start with the oral presentation.
Those who are in session A and B, please leave session C and you can join session A and B, that is for biological sciences. Please join that session. The link has been provided to you in your WhatsApp group.
Shall we start?
Yes, Bhumi, you can start with the session. Is the participant is there? Oh, sorry, I'm sorry. Is it C session? Benazir, it is C session,
Yes,
no?
sir.
Okay, okay, I'm
Yes,
sorry.
yes, sir. Yes, sir.
So now let us start with our oral presentation session. So now I request Professor Vijay Desle sir to introduce chairperson for today's oral presentation session, Professor Shivram Garje sir, Head of the Chemistry Department, University of Mumbai. Over to you, sir.
Thank you, madam. It is great privilege for me to introduce Professor Shivram Garje sir. Sir has completed his MSc in Inorganic Chemistry from University of Mumbai in 1993. Sir has completed his PhD in Organometallic Chemistry from Baba Atomic Research Center, Mumbai, with Dr. V.K. Jain in 1997. Sir has completed his postdoctoral research in nanomaterials from University of Manchester, UK, in 2005-6. After completing MSc, Sir has joined Quality Control Department in US Vitamins at Mumbai in 1993. Then Sir worked as a lecturer in inorganic chemistry in the Department of Chemistry, University of Pune in 1998. Then Sir joined lecturer as a lecturer in inorganic chemistry in the Department of Chemistry in Institute of Science in 1999. Then Sir joined lecturer in inorganic chemistry in the Department of Chemistry from January 2000 in University of Mumbai. Then Sir became professor in chemistry at the Department of Chemistry, University of Mumbai from March 2014. Some prestigious positions held by Sir are: Sir was in-charge director of University of Mumbai's Garwari Institute of Career Education and Development from April 2019 to 2022. Sir was also in-charge director of Western Regional Instrumentation Center of University of Mumbai. Then Sir was also in-charge director of National Center for Nanoscience and Nanotechnology, University of Mumbai, from 2021. Then Sir was also served as an associate dean in Science and Technology of University of Mumbai. Now Sir is serving as the head of the Department of Chemistry, University of Mumbai. Sir is a member of syllabus committee. Sir is a member of RRC, Board of Studies. Hence, some of the important awards received by Sir are: in 2002, Sir received Young Scientist Research Award from the Department of Atomic Energy, Government of India. Sir received Boy Scout Fellowship from the Department of Science and Technology, Government of India. Sir has also received Performance-Based Incentive Award from University of Mumbai. Sir's membership in the professional societies are: Sir is a life member of Indian Chemical Society. Sir is a life member of Indian Association of Chemistry Teachers, life member of Material Research Society of India. Sir is a member of Royal Society of Chemistry, member of American Chemical Society, Indian Society of Material Chemistry, Chemical Research Society of India. Sir is Executive Council member of Material Research Society of India, Executive Council member of Indian Chemical Society, Joint Secretary of Indian Chemical Society, Vice President of Indian Chemical Society, Mumbai chapter. Sir has published more than 65 research papers. Sir has completed 3 research projects, and Sir has more than 1100 citations. Sir has also presented 86 papers in the conferences. Research areas of Sir are material chemistry, designing molecular precursors of nanoparticles. 10 PhD students completed under the guidance of Sir. 4 students are working under his guidance. 2 students completed postdoctorate under the guidance of Sir. Now I request Professor Garje sir to take a charge as the chairperson of the session. Over to you, sir.
Thank you very much, sir. Am I audible?
Yes, sir. Yes, sir.
Okay. At the onset, I thank Principal Kulkarni Madam, Dr. Nirgode, and all the organizing team of this particular conference for giving me opportunity to chair this particular session. And as mentioned earlier by Benjin Madam, there are 5 minutes for each presentation, and we can have maximum 2 questions for a particular presentation. In this session, we are going to have 24 oral presentations. So one by one, I will invite the speakers, and then they can present their presentations in 5 minutes, which will be followed by 2 questions from the participants. And if I have any query, I will be also asking maybe a single question also. So first presentation which we have is OC1 from Mr. Dilip Kumar Yadav from the KJ Somaya College. The title of paper is Efficient Method for Formation of Carbon-Nitrogen Bond Moieties. So are you there, Mr. Dilip? OC1, Mr. Dilip, please come online and start. If you're there online.
Dr. Dilip, are you there? Dr. Dilip Yadav?
If not there, then we can go ahead with next one and we can later on give him a chance. So let us go ahead with the second one and we can come back always to the first speaker if he becomes available later on. So second presentation is by Dr. Archana Singh. She's from Birla College, and the title of her paper is Effect of Dopant on Electrical Properties of Ternary Characterized Compounds. So over to you, Archana. I can see her online. So Archana madam, you can start the presentation.
Dr. Archana Singh, are you there? Please start with your presentation.
She was there sometime back.
Dr. Archana Singh, you are not audible.
Yeah, Archana madam, you share your presentation and you can start.
Madam, please start with your presentation.
I think she got disconnected.
Yeah, I think so.
Dr. A.S. Patil is there from.
May I audible, sir?
Yeah, are you there?
Yes, sir.
Yeah. So what we will do is we can have your presentation first, and
Yes,
meanwhile,
sir.
if the other speakers they join, then we can take up them.
No problem, sir.
Okay. So thank you for being with us. Dr. A.S. Patil is there from Dr. P.R. Gogre College, Science College, Dhule. And the title of his paper is Advanced Oil-Based Microcapsule Bridge Formulation of Zinc Metal Sulfide and Phosphide with BSc for Multifunctional Agricultural Applications. So over to you, Dr. A.S. Patil.
Its screen is visible.
Yeah, you can make it like on presentation board. Just press the cup on the lower side here. Yeah.
Its screen visible, sir.
Screen is visible, but you can make it on the presentation board. This, yeah, now it is okay. So please start. And madam, you can keep the time, Benjamin, madam. Someone you can keep it. Yeah, please start.
Good afternoon, everyone. Myself, Anil Patil. My research title is Advanced Oil-Based Microcapsule Bridge Formulation of Zinc Metal, Metal Sulfide, and Metal Phosphate with BSc for Multifunctional Agricultural Application. I am doing this research work at Department Chemistry, SSVPS Sanstha's LK Dr. P.R. Kogre Science College, Dhude, Maharashtra. My research objective are to make easy and efficient formulation using advanced to prepare multifunctional formulation for agricultural application means it act as a pesticide or it act as a fertilizer or plant growth regulator, that is all in one or what all purposes. In microcapsulation, core material is covered with cell material, which prevent environmental degradation of active ingredient. In some cases, pest and insect has developed resistant power against pesticide and insecticide. This happens due to excessive use of pesticide or agrochemical. Uh, the unique application of control is of active ingredient increase the efficiency and efficiency of agrochemical. Microcapsules are water insoluble. They are remains long time on crop and reduce the aquatic toxicity and volatilization of active ingredient. Our focus to replace synthetic pesticide by other pesticide having natural origin, that is neem oil uh, or or peroxide. Mainly and efficient and economically beneficial agricultural practices for farmer. Microcapsules were prepared in my laboratory by 50 ml 5% polyvinyl whole, 50 ml water, 3.38 gram urea, zero point 0.1 gram sodium lauryl sulfate in 250 ml from bottom plus. Reaction mixture was stirred.
Sir, if you are changing the slides, yeah, now it is okay.
Yes. Okay. At 600 RPM for 15 minutes and pH is adjusted above 7. Homogeneous mixture of 9 gram oil and 1 gram active ingredient. Active ingredient is zinc, zinc sulfate, zinc phosphate, BSC was added dropwise with stirring about 700 RPM. After 30 minutes, 10 gram formaldehyde was added and reaction is stirred at 700 RPM and heating is started. Temperature was kept constant at 65 degrees Celsius and pH were adjusted below 4. After 30 minutes, 0.5 gram resinol were added to reaction mixture and stir at 700 RPM for next 3 hours, and finally microcapsule are filtered out. Primary level formation of microcapsule is characterized by optical electronic microscope. These are the images of optical electronic microscope. In gray, zinc is encapsulated.
Slides are not changing.
Already changed, sir.
Yeah, now it is okay.
Okay. In figure A, zinc is encapsulated. Zinc metal as encapsulated. In figure B, zinc sulfate is encapsulated, and zinc in figure C, zinc phosphate is encapsulated. In D, BSC is encapsulated, and figure E, zinc sulfate and BHC is encapsulated. Variation in size is observed here, and it is by using scanning electronic microscope. These are the images of scanning electron microscope. Stability of a microcapsule is studied by thermal grammatic analysis. These are the stability and it is indicate the formation of microcapsule as okay. Okay. I would like to thank the organizing team for giving opportunity for me to present here my research work.
No, you can show your
Any
conclusion. Yeah, conclusion you can show us.
Okay. Okay. Yes, sir. The microcapsulation of zinc as a zinc metal, zinc as a zinc sulfate, and zinc as zinc chloride are beads with BHC, benzene chloride, is most efficient way of formulation as well as increase efficiency and durability of agrochemical. This is the conclusion, and these are the references.
Okay, are there any questions from the participants? Okay, sir, one question. So these
Yes, sir.
agrochemicals, microemulsions, what is their application you say?
Application is to encapsulate using neem oil, means natural oil and synthetic insecticide. We try to both mix and encapsulate them.
And then their further application will be as something like
Sir, yes, sir. Yes, sir. Further application, we already under progress. Already we study on cotton crop.
So it will be something like pesticide or something like that you are saying.
Oh, yes, sir.
Why I'm asking is that if it is encapsulated, then how
yes.
easy it is to release and have that effect as the pure chemicals. Because pure chemicals they can straightway act as pesticides, but if these are the encapsulated ones, then how the release will be there and how effective your products will be as agrochemicals.
It is a unique property of microcapsulation, time bound release. Ingredient is released in time interval. That's why no need for multiple doses to agrochemical.
Okay. So can we consider that as an advantage?
Yes, sir.
Okay, thank you very much,
Okay.
Dr. Anil Patil. Any question from the participants? Okay, if there are no more questions, let us thank Mr. Patil for his nice presentation. Now we move on.
I think so. Dr. Archana Singh is available now.
Yeah. So, Archana madam, can you start with your presentation? Yes, is there, right? Archana Singh madam from Birla College. I think some problem with her net. Mr. G.S. Sonavni is there.
Yes, sir.
Okay. So we can start with your presentation and I can go back to our earlier one if they are there. So now we'll have a presentation which is OC4 and that is by Mr. G.S. Sonavni. I'm sorry if I'm pronouncing wrong as a doctor or Mr. I do not know whether these are the research scholars or faculty members. So pardon me for that. So yeah, Mr. Sonavni is from Kisan Arts, Commerce and Science College, Parola, Jalgaon. And the title of his talk is Hydrothermal Synthesis of Activated Carbon Immobilized WO3 nanoplates, synergistic effect of adjacent photocatalytic catalyst for degradation of organics. So over to you, Mr. Sonavni.
Thank you, sir. Sir, is my screen is visible?
Yeah. And you can go on presentation mode.
Okay. Thank you, sir. Yes. Good afternoon, one and all. Respected Chairperson, Professor Garja sir,
all the dignitaries on the virtual conference. First of all, I would like to acknowledge my sincere thanks for Mehta College for giving me this opportunity to present my work over here. Myself, Dr. Gunwant Sonone, from Kisan Arts Commerce Science College, Parola, District Jalgaon. Today's my work, which work I am going to present over here, hydrothermal synthesis of activated carbon immobilized tungsten oxide nanoplates, synergetic effect of adsorption photocatalysis for degradation of organics. We know that there is increase of industrial and agricultural activities in every developing countries. Every activity uses high synthetic chemicals. These synthetic chemicals are toxic in aquatic environment. It has been observed that 80% of the diseases are directly related with the polluted water. There are different methods to remove these. Conventional methods are there, chemical, physical, biological, but each method has its own limitation and advantages. Few of the limitations are, most of the method, there is only transfer of pollutant from one phase to the another phase, ineffective towards the complete removal because of the stability of the regeneration of matter and economically ineffectiveness. These are the major drawbacks. Hence, the solution for this problem is heterogeneous photocatalysis. Photocatalysis is one of the advanced oxidation process. These are environment friendly and also green technology, green chemistry approach, both applicable in aqueous as well as in gaseous state. In this, solar energy is used as a light source, so economical viable process. During this, what happens? When the beam of light interacts on the photocatalysis, the electron shifts from valence band to the conduction band, and electron interact with the oxygen of the conduction band, forming oxygen peroxide, while the whole interacts with the water molecule, releasing OH radical, and these radical interacts with the organic pollutant and carry out the degradation. It has different advantages also, but we are looking toward the synergy of adsorption and photocatalysis. Majority of the photocatalysis are found to be effective only in UV light. Hence, we need to use in the visible region. We are making the combination of both processes as adsorption and photocatalysis. So the system is called as integrated photocatalytic adsorbent. The synergetic combination does not only preserve all interesting characteristics of both individual components, but also overcome the drawbacks of these processes. And hence, these processes are economically viable. We have selected tungsten oxide as an photocatalysis because of its desired band gap, suitable morphology, high surface area.
Extremely sorry to interrupt, but your presentation is not visible, sir.
Oh.
No, madam, it's there.
Is it visible, sir?
Yeah, it is. Madam, you can pin his presentation.
Yeah, you can pin
Okay,
it.
okay. Sorry, sir. You can continue.
Okay, thank
Please
you.
continue.
The experimental setup we have used, method we have used is hydrothermal process. These are what the Teflon container over which we have synthesized this material. The method is solvothermal, as we have used organic solvent rather than water. This is what the experimental setup, where we have used visible light as well as the UV light for the external process. This is what the method for the extraction of the eluent from the synthetic dye material. The spectrophotometer is used for this analysis. This is what the process where a typical synthesis of this tungsten oxide is carried. This is the reported method. This is what the hydrothermal autoclave Teflon bottle which I used for this process. Now results and discussion. The material we have synthesized is activated carbon loaded tungsten oxide. So the XRD of the pure activated carbon, XRD for the tungsten oxide, and the activated carbon tungsten oxide was done. These are what the different peaks reported, corresponds to the different planes 0, 1, 0, 1, 2, 0, 1, 2, 0, 1, 1, and so on. This confirms that the material which we have synthesized is having orthorhombic structure and also supported the JCPDS data. We have also carried out the structural, measured the structural parameters. Structural parameters shows the different crystalline size in the nanometer range and also shows the negative strain. The negative strain may be because of the excess of addition of the activated carbon. These are the same images which shows that the formation of the nano tungsten oxide plates having nanometer size, pure tungsten oxide, then tungsten oxide loaded with the activated carbon in different proportion 40, 60 and 80 milligram per liter. Now the elemental composition supports the presence of tungsten as well as oxygen. The same images supports the formation of the nano tungsten oxide plates. Optical analysis was used to evaluate the band gap of the synthesized material, which is an important property of this one. The band gap is 2.31, 2.28, 1.90, 1.88 for the different composition, and hence they are used for the photocatalysis. Thermogravimetric analysis or TG analysis supports the stability of the material, pure, as well as the all other activated carbon-loaded tungsten oxide. The textural properties are also evaluated to find out the pore size. We also called as a BET analysis was carried out. The pore size, pore volume, micropore and mesopore was find out. The external surface area for activated carbon-loaded tungsten oxide for AT is found to be 77.359 meters square per gram. This support that also it has been found that these material which we have synthesized are mesoporous and multimodal micropores. Adsorption isotherm, the supported we have carried out for Friendly's and Langmuir. It supports the Langmuir adsorption, and hence it is chemo type of the adsorption. Homogeneous. This is because of the homogeneous distribution of active site on the activated carbon-loaded. Yeah, within a minute I will conclude. The calculated QV value are supported with the experimental one, and hence follows pseudo. Also, follows the pseudo-second order kinetics. We have carried out the photocatalysis in dark as well as in presence of the light. But when the activated carbon-loaded tungsten oxide was carried out, it supports that with the increase in the proportion, the percentage degradation was increased from 41.62 to adsorption, photocatalysis 46, while the combined effect was 88.59. So I conclude that the morphology of this material was find out, which are mesomorphous tungsten oxide nanoplates, which are homogeneously dispersed. It follows Langmuir adsorption and pseudo second order carried out in visible light. So increased adsorption along with the decrease in the photocatalytic efficiency of activated carbon tungsten oxide nanocomposite is mainly due to the increased adsorption along with the decrease in recombination of the photo-generated electron hole pair of this one, and hence it is viable. So these are what the few conclusions we have drawn from another systems also we have carried out, and these materials are more suitable for this one. So this article we have published in Inorganic Chemistry Communication with my research scholars, Mr. Dr. Labane and Dr. K.S. Bausar, with my research colleague, Dr. Arbitek. I acknowledge my sincere thanks to Instrumentation Center, as well as School of Advanced Sciences, VIT Vellore, and School of Chemical Sciences. Thank you. Thank you.
Okay, thank you, sir. Very excellent presentation. So first we can take a few questions if participants have. Participants, any questions? Okay, yeah, maybe I can ask few queries. Have you compared the band gaps of pure tungsten oxide and the composite comparatively?
Yes, sir. Yes, sir.
So what is the difference?
Shifting was observed in the band gap from 2.88 to 1.83 electron volt.
Okay. Because basic objective of your work is to shift band gap towards visible region probably.
Yes, sir. Yes, sir. Yes, sir.
So that you can use it as
yeah, in the visible region.
Visible region, yeah.
Yes, sir. Solar light can be used.
Yeah, yeah. And efficiency wise also, have you seen the difference?
Yes,
just
sir.
take the pure tungsten
We are,
oxide
yeah. Actually, we are expecting synergetic effect, but it is not that much as per our expectation. It is slightly increased actually that we have reported because of adsorption, because of photocatalysis, and when we carried out in combination. So it should not be additive effect. Our expectation is it is synergetic. So it is not full synergetic and full additive actually. It is in between that one.
So that is reflected in the band gap variation. The
Yeah.
band gap is not much shifted.
Yeah. That's what it is.
And you can also couple your study, like take the final product and see whether it is decomposed into maybe water and CO2, that kind of thing.
Yeah, we have carried out, sir. We have also find out the mechanism, scavenging study and all the related, but as per the limitation of time, I have taken that much work.
So nice work, sir. All the best. Thank you very much.
Thank you. Thank you. Thank you.
Thank you. Okay, so now we move on to the next presentation. So, Archana madam, are you ready or still struggling? Archana Singh madam. Maybe we can take up the remaining people at the end. So we have OC5 and that is by Namrata Khandekar. So Namrata Khandekar madam, are you there?
sir.
Okay. So we'll have this presentation first now, which is OC5, and she is from the Department of Chemistry, University of Mumbai. How come I have not met you? Anyway, no problem. So the title of her talk is Development and Synthesis of Novel Chalcone Analogs derived from 2,3-dihydro 1H naphtho 2,1B pyron 1,1. So over to you, Namrata.
Sir, screen is visible.
Yeah, it is visible, and you can go to presentation mode.
So whether it is fine.
You can zoom it out a bit.
Yeah. Is it PDF format, not PPT?
PPT.
So single slide you should show. I think you are doing it on mobile. Yeah, this is okay, the earlier one.
Yes, sir. I am doing it on that, sir.
So you hold it horizontally and then you can do it. Okay, I think that will work. Go ahead with. Yeah, this is okay.
So good afternoon, everyone. Good afternoon to our respected Professor Shivam Garje. So myself, Namrata Khandekar, and my topic is development and synthesis of chalcone analogs derived from 2,3-dihydro 1H naphtho 2,1B pyrene 1 on. So these are the contents of my topic. Introduction. Chalcones are basically two aromatic rings which are linked by an aliphatic 3-carbon chain, and this 3-carbon chain is keto-ethanolic group. Previous work: to obtain chalcone, 2-hydroxyacetophenone reacted with benzaldehyde in the presence of base NaOH, and this reaction goes by Claisen-Smith reaction to obtain chalcone. For present work, synthesis of novel using precursor 2,3-dihydro 1H naphtho 2,1B pyrene 1 was prepared using was synthesized using acid catalyst and base catalyst. Experimental work: beta naphthol was reacted with acronyl nitrile with triethylamine, which further gave the adduct, and by hydrolysis of this adduct we obtained our desired precursor. So H1 NMR spectrum of precursor: triplet proton peak, which is of CH2, is obtained at 3 ppm, and triplet proton peak, which is of CH2, is obtained at 4.5 ppm, and between 7 to 8 ppm aromatic protons has been obtained. 13C NMR carbonyl carbon peak, which is obtained at 196 ppm, and aromatic peaks are between 110 to 137 ppm. IR spectrum of precursor: peak at 1657 centimeter inverse carbonyl carbon peak has been obtained, and CO stretch peak has been obtained at 1207 centimeter inverse. So with the help of precursor, this is our main reaction scheme, and with the help of precursor we have obtained an chalcone analog it was reacted with derivative of benzaldehyde in the presence of base NaOH and ethyl alcohol, and this reaction was taken place at room temperature for 12 to 10 hours. So this is H1 NMR spectrum of the final product, that is chalcone analog. Around 5.5 ppm aliphatic proton peak has been obtained, which confirms the formation of chalcone analogs. And around 7 to 8 ppm aromatic protons are obtained. 13C NMR spectrum, around 196 ppm carbonyl carbon peak has been obtained, and around 138 ppm aliphatic carbon peak has obtained, and around 120 to 140 ppm aromatic carbon peak has been obtained. So this is the IR spectrum. Carbonyl carbon, which is obtained at 1614 centimeter inverse peak of carbonyl carbon is obtained, and CO stretch, which is obtained at 1200, and C aliphatic C double bond C stretch has been obtained at 1654. So comparison of the obtained yield. So by using acid catalyst, high percentage of yield was obtained, and also the concentration of acid catalyst affect the percentage of yield. So we conclude that by using acid catalyst, high percentage of yield was obtained, but also the advantage of using acid catalyst is that it was solvent free. So these were the references used and acknowledgement. I could like to thank to Professor Shivram Garje, the head of the chemistry department of university, and RG Thorat sir, Mr. RG Thorat sir, to giving me the opportunity to work under him. And also I could like to thank our conference committee for giving me opportunity to present my work here. Thank you.
Okay, so thanks, Namrata. I suppose this is your project topic, isn't it?
Yes, sir.
Okay. So now tell me that what is the importance of chalcones? Why you have chosen?
The chalcones are basically shows biodynamic activities. So their importance is actually they show biodynamic activities and can be used for disorders like anti-malarial or anti-inflammatory for pharmaceutical medicines.
So you meant to say that they can be starting materials to formulate drugs and those kind of things. That's what you want to
Sir,
say?
can you please? Sir,
They can act as a starting materials for the synthesis of drugs.
Yes, they can be used in many disorders for in pharmaceutical as a medicinal purpose in medicinal purpose.
Okay. Now one more question is that you said that acid catalyst you get more yield. So why acid catalyst reactions are giving more yields?
Sir, acid catalyst give more product because of the steric in acid catalyst, as well as their resistance also affect inductive effect also.
Anyway, in your project you look into all these things, okay? Like why the acid is enhancing the rate of reaction, what is the mechanism and all this things. Okay, so all the best, Namrata. Anyway, before that, any participant want to ask any question? Okay, Namrata, you can now close your presentation and keep on listening to others.
One of the objectives of us.
Okay. Now, Archana Madam, you are trying to present your presentation. Are you ready? Okay, now, Sneha Madam, kindly mute your mic. Sneha Madam. Other participants kindly mute the mic. Naid Sheikh, are you there? Naid. Naid, are you there? आपकी आवाज क्लियर नहीं आ रही.
Yes, sir.
Okay, okay. Okay, so now we have next presentation, that is OC6, synthesis of partially saturated pentrahydrohelicin by Naid Sheikh, and she's from Department of Chemistry, University of Mumbai. So over to you, Naid.
So is my screen visible?
Yeah. Perfect. Go ahead.
Good afternoon, everyone. My name is Sheikh Nahid Sadiq, and the topic of my presentation is synthesis of partially saturated pentaheterohelicins. These are the contents discussed in the presentations. Helicins are auto-condensed polycyclic aromatic compounds in which benzene rings are angularly attached to give helically shaped chiral molecules. Now, helicins are of two types: carbohelicins, in which only carbon skeleton is present, and heterohelicins, in which along with carbon skeleton a heteroatom is present. So, these helicins can be prepared by various methods, including photocyclization, Diels-Alder reaction, Friedel-Crafts reaction, metal catalyzed cyclization, etc. In our present work, we try to synthesize oxahelicins by using Diels-Alder reaction. So, it consists of 4 steps. In first step, phenol undergoes microaddition reaction with acrylonitrile to give you compound 2. Now this compound 2, the cyanide group present in compound 2 undergoes hydrolysis followed by cyclization to give you compound 3. Further, this compound 3 couples with itself to give you compound 4, which acts as our diene. That diene undergoes Diels-Alder 4+2 Diels-Alder reaction with benzene to give you to give our product, which is oxa 5 helicin. Now the the formation of the desired product is confirmed by spectroscopic methods. In 1H spectra of compound 2, you can see two ranges. We can see peaks of aromatic regions and de-shielded aliphatic region because of presence of cyanide and ether functionality. As for IR spectroscopy, you can see regions for aromatic regions, and the presence of cyanide group is confirmed by peak at 2200 centimeter inverse, followed by carbon oxygen stretching at 1225, which is characteristic peak of ether functionality. As for compound 3, you can see in NMR spectrum that you have two regions, aliphatic as well as de-shielded aliphatic as well as aromatic regions. As for IR spectrum, you can see bands for aromatic region, aliphatic region, followed by presence of peak at around 1700 confirms the presence of ketonic functionality, and carbon oxygen stretching band at around 1225 centimeter inverse. Compound, as for compound 4, its 1H spectrum shows de-shielded aliphatic region and aromatic region, followed by your IR spectra. In IR spectra, you can see presence of aromatic region, which is present at around 3000 centimeter inverse, which is carbon hydrogen sp2 hybridized stretching, followed by you have a peak of ether functionality at 1223 centimeter inverse. For our main compound, which is helicins, in NMR spectra, we can see de-shielded aliphatic region as well as aromatic region. For IR spectrum, we can see the peaks at 3000 centimeter inverse for carbon hydrogen sp2 hybridized stretching, followed by 1600, the peak at 1600 for carbon double bond carbon stretching, followed by your ether linkage, which is at 25 centimeter adverse. So I would like to conclude here. We synthesized oxahelicins by using lab reagents, and different phenols, substituted phenols can be used to get our substituted penta-oxahelicins. These are the references I used. Thank you. I would like to thank Dr. Shivram Garje, HOD, Mumbai University, Mr. R.G. Thorat, our microanalytical staff, and NB Mehta College for giving me an opportunity. Thank you so much.
Okay, thank you, Naid. The participants, any question? Okay, so Naid, just two queries. What is percentage yield of this compounds?
Percentage yield, sir, was about 70% yield after doing column chromatography.
You are getting quite high, as means they are somewhat sterically hindered compound. So still, you are getting quite good yields. Okay. Okay, nice work. Thank you very much, Naid. Thanks. Any question from anyone else? If not, let us go to next one. So EMS Tiwari is there?
Yes, sir.
Zero, ma'am, am I audible?
Sorry?
Am I audible, sir?
Acha, Archana madam. Yeah, you are audible.
I'll give after him.
Okay, okay. So let Tiwari sir give the presentation. So now we have the Tiwari, I think he's student.
Yes, Mrityunjay Tiwari, sir.
Okay, okay.
Thank you. Thanks a lot. Thanks a lot.
Wait. Let me. So this is OC7 and the title is Determination of doxycycline in human body fluids using modified screen printed carbon electrodes. So he is again from the University Department of Chemistry, Mr. EMS Tiwari. So now, Mrityunjay, you can start your presentation.
Thanks a lot, sir. Thank you for the introduction. Thank you for the organizing committee to for accepting the proposal of my oral presentation. I think my screen is visible to everyone.
Yes, yes.
Yeah. So myself, Dr. Mrityunjay Tiwari, I am pursuing the PhD under the guidance of Dr. Varun Kadu. And the topic for the presentation is determination of doxycycline in human body fluids using modified screen printed carbon electrodes. These are the following bullet points which will be majorly discussed as we go ahead with the presentation. So as we know, majority of the human health depends upon the food products what they consume. Besides of the beneficial constituents in the food products may also contain some contaminants and some antibiotic residues which are being used while the processing and the pre-processing part. So and due to the over demand of these food products in the market, these products, these antibiotic drugs and these products are extensively used for the to meet the huge amount of production limit. One such example is use of doxycycline, which is having a broad spectrum antibiotic towards gram-negative and gram-positive bacteria and has been extensively used in animal breeding. This doxycycline has also been used as a specific treatment against COVID-19 in India and Brazil due to its in vitro activity against SARS-CoV-2. Hence, monitoring of doxycycline in food and body fluid is meaningful due to the abuse of antibiotic that leads to serious health problems and bacterial resistance. Over here, we have developed used a two-step reduction method to develop our modified sensors, wherein graphene oxide, which was synthesized using modified Hermes method, was electrochemically reduced using cyclic voltammetry. You can see the graph over here, figure 1. This is the graph representing the cyclic voltammetric deposition of graphene oxide to form the ERG SPC, that is electrochemically reduced graphene oxide modified screen printed carbon electrode. Followed by the deposition of gold, which was achieved by again using cyclic voltammetry in the range of 0 to 1.2 volt in 1 millimolar acidic solution of orochloric acid. So after the electrode has been modified, we have modified 3 different types of electrodes.
The one which was like AU-ERG-SPC, that is our finally modified gold electrochemically reduced graphene oxide and screen printed carbon electrode. Other one was SPC, that is the bare SPC without any modification. The third one was AU-SPC, and the fourth one was ERG-SPC. As we can see, over here we have done electrochemical characterization using 5 millimolar of redox mixture, that is a ferroferricyanide in equal molar ratio in a solution of 0.5 molar KCl. And over here, if we can see the graph, the peak current for oxidation and reduction is greatest for the final modified electrode, and that the same can be depicted from the EIS curve. So after the electrochemical calibration, we have performed the pH optimization. As we know, pH plays a very important role for electrochemical oxidation or reduction of any molecule. So over here we have used Bitter-Robinson buffer in the pH range of 2.5 to 7, containing 50 micromolar of the analyte drug, and we have done a pH optimization using square wave voltammetric technique. And from the graph 5 and 6, we can see at pH 3 we are getting a maximum peak current and at a proper peak shape, and a dumbbell shape peak. So it signifies that at acidic medium is supportive medium for the oxidation of doxycycline electrochemically. We have also performed a scan study, and the results are like this. Like, as the scan rate is increased from 20 millivolt to 200 millivolt, we are experiencing a linear increase in the oxidation as well as reduction peak current, which can be even seen in the graph, like the square over here. Okay. So after that, we have done the electrocatalytic activity of the modified electrode over the screen of our modified carbon electrode. So we have used a cyclic voltammetry as well as square wave voltammetry to verify the oxidation of doxycycline over our modified electrodes. As we can see, in both the techniques, AU-ERG-SPC, that is the final modified electrode, is giving better peak current and at a better peak shape as compared to the other modified electrodes. The calibration curve was done in the concentration range of 10 to 100 micromolar, and the LOD detected was 4.28 micromolar per liter with an LOQ of 14.3 micromolar per liter, and the R square value for the calibration curve was generated to be 0.993. Sensitivity of the electrode was calculated to be 0.194 micromolar per centimeter square. After this calibration curve, we have spiked the doxycycline of T3 varying concentration 10 micromolar per liter, 30 micromolar per liter, and 50 micromolar per liter in human blood, healthy human blood serum. And the process was like this: we have taken a blood serum and we have diluted it with 10 times of the BR buffer, and we have spiked 3 different concentrations, and we have done the analysis of the spiked serum samples. So the recovery study was found to be in the range of 94.3 to 99.6%, indicating an excellent activity of modified sensors for real matrix study. In conclusion, we have developed a low-cost disposable sensor based on a screen-printed carbon electrode modified with AU-ERG-SPC using electrode reduction method. The synthesized sensor consists of stacked ERGO sheets and homogeneously dispersed gold, showed the large surface area, high electrical conductivity, great charge transport capability, and electrocatalytic activity for oxidation of doxycycline. The sensor exhibited desirable analytical properties towards doxycycline, having wider linear range with high sensitivity, a low detection limit, and great visibility for its use in food products and blood serum with acceptable accuracy and recovery. These are the following references from where we have taken the idea of the development of sensor and the drug molecule which is to be used and used based on the current hot topic of COVID. This drug was widely used in the entire world for against its activity. Thank you.
Thank you, Mrutyunjay. Very excellent work and excellent presentation as well. Participants, any questions? Okay, so just few queries from my side. First thing is that you are saying that when you optimize for peers, then at PS3 it is better.
Yes, sir.
So then if you want to carry out in vivo studies,
Yeah.
you want to develop a sensors where
Yes.
actually in vivo it can be used. So can we use your say you fabricated device? Can we use it?
Currently, at this moment, the research is mostly developed on the PS3 itself. For having a better LOD, we have optimized it at PS3. If we take this research further, and we can get a point where there are n number of reports available on the on the websites where they have reported at pH 7, which is the normal pH of the neutral pH.
Yes. So in your research, you try to bring your sensor optimized at PS7
Definitely,
with the
sir.
modification that way, so that maybe you can actually fabricate a device and then it can be used.
Yeah,
Yeah.
definitely.
Uh,
And that will be tried for sure, sir.
and one more thing, is it selective? Say you are other.
Yeah, sir. We have, yeah, based on the different biomolecules and different target analytes. Like when we take a blood serum, most of the salts are present, like ions are present in nitrite ions or magnesium ions. So we have done the selectivity study over there. And the selective study is based on the pH at least. So at pH 3, we are not getting any interference from the other molecules present in the serum.
And you are saying your modified electrode is giving better sensitivity. So what is the reason for that?
Sir, it is because of the high electrical conductivity for sure. Because when we compare to screen printed carbon electrode and for maybe even with the graphene electrode, the conductivity is definitely increased in case when we are modifying with the gold particles.
Okay. And can we assign some part to the increase in surface area or something like that?
Increasing surface area, sir. We have. I'd say I didn't get your question, sir.
No, the sensitivity increases. You are saying it is increase in the conductivity.
Correct.
So my point is whether the increase in surface area in the composite or in the modified electrode, that is also responsible.
Definitely, sir. Yeah.
So I mean, you try to correlate all those things. Okay.
Yes, sir. Definitely. Yeah. Yeah.
Okay. All the best, Mrutunjay. Thank you for your presentation.
Thanks a lot. Thank you, sir.
Now Archana madam is there. So
Yes,
Archana
sir.
madam, yeah. So welcome madam, and
Yes, sir.
she's from Birla College, and her title is Effect of Dopant on Electrical Properties of Ternary Charginate Compounds. It is oral presentation number 2. So
Okay.
madam, over to you.
Okay. Very good afternoon, sir. Outline of my presentation consists of introduction to chalcogenide compounds, synthesis of ternary chalcogenides, SBSI and SBTI, suitability of the method for doping, electrical properties of SBSI and SBTI, effect of dopant on the electrical properties of materials, and conclusion. The ternary chalcogenide materials of group 15, 16, 17 have attracted the minds of researchers due to their wide range of potentially useful and interesting properties. In our lab, we have synthesized SBSI and SBPI by solid-state thermal reactants by taking a stoichiometric ratio of constituent elements in a quartz tube, which was evacuated and sealed under vacuum and heated up to 500 degrees centigrade for 4 to 5 hours. This was the pic of the evacuated tube in which we have taken our constituent elements in the stoichiometric ratio, and after certain type heating at certain temperature, we got gray shiny crystals were obtained in the form of bunches, and the morphology of the bunches of polycrystalline needle-shaped crystals were characterized by EDEX and SEM. The XRD, SEM, and EDEX characterization were confirmed the formation of these compounds. Later on, we studied the electrical behavior of the compounds by two prop setup and Hall prop setup, and these apparatus were used to measure the capacitance. These are the Hall prop setup and two probe setup developed at our NTRC Birla College, and this one, the third one, is the LCRQ meter device, which was used to measure the capacitance. Variation of resistance of sample at different temperature was studied for constant current And it was found that first resistance increases with increase in the temperature till 300 Kelvin, and then it becomes constant. And thereafter, an interesting behavior we found that at higher temperature, its resistivity abruptly decreases. The other parameters were also determined as a part of electrical charge density, carrier mobility, and dielectric constant. The lattice parameters are very close to reported data, and the intensities and position of the uh, peaks are in a good agreement with the literature values. In the same way, result of the electrical properties of ternary chalcogenide SBTI also observed, and this graph shows the variation in the resistance versus temperature of SBTI sample in the temperature range of 4 Kelvin to 300 Kelvin. And again, we observe the same conducting behavior of these materials. Charge carrier density, carrier mobility, and dielectric constant was also measured with one of my colleague from Department of Physics, Dr. Harish Dubey sir. And then doping of the compound was done by introducing tin and indium. The doped compound were also synthesized with similar parameter as that of the pure SBSI and SBTI compounds, and quaternary compounds were synthesized using solid state thermal reaction techniques. These are the different elements in percentage of elements with the dopants. Table gives the value of resistivity, conductivity, activation energy, band gap, and dielectric constant of the sample prepared by doping SBSI and SBTI with tin and indium, and the value of the resistance of doped materials obtained were found to be high in the range of 2000. This table also compares the value of resistivity by the two-probe method and comparison of the values of resistivity of the doped compounds obtained in the presence and absence of inert atmosphere. And it has been found that displacement of sulfur atom by indium atom does not have much impact on the SBSI, but activation energy decreased drastically compared to decrease in the resistivity at room temperature. And it has been observed that there is decrease in the dielectric constant due to the decrease in the polarizability. The replacement of 50% sulfur by tin atom also has similar effect on the resistivity as well as conductivity. And the doping of tin at the antimony site gives much better improvement in the conductivity. Doping of tin at the tellurium site of the SBTI gives improvement in the conductivity of the primary materials. Therefore, tin doped at the tellurium site of SBTI gives better results in all the respect, and still our work is going on in this field. We can conclude that solid state thermal reaction is a suitable method for synthesis of chalcogenide materials, and SBTI is also a suitable method for doping of a material. The conductivity of the dope antimony sulfur iodide can be improved by doping it with tin or indium, and the conductivity of tin doped SBTI is higher as compared to the pure one. The band gap and activation energy can be reduced by doping tin and indium in SBSI and SBTI. I must thank University of Mumbai for sanctioning our project choice for this work. With their help, we can do something. And I'm thankful to BK Birla College, Kalyan, NTRC Lab, and Department of Physics and Department of Chemistry for conducting my work. Thank you so much, organizers, for giving me opportunity to participate.
Thank you, Archana Madam. Participants, any question?
Yes, sir.
Yeah, please ask. Guru sir.
To measure mobility, we need to calculate thermal conductivity as well as electrical conductivity.
Yes, sir.
How you have measured both thermal as well as electrical conductivity?
We have mobility, that conductivity rho L upon E by using and
No,
activation.
that is true. It is nothing but the ratio of thermal conductivity to electrical conductivity.
All from setup.
Experimentally, how you have measured?
Yes, sir. How by using
thermal as well as
She's saying Hall measurement.
all from setup. All from setup.
Okay.
high probe method.
Any other? Okay, just two queries from my side, Archana madam. That quaternary composite, you said you have shown some percentage elemental composition. How that was calculated?
The quaternary, just a moment.
Sorry.
This one, quaternary. What I have replaced one of the constituent, that SBSI, that is some percentage of S was substituted by indium, and some of the S was substituted by this one
No,
tin.
no, that is okay. The next one, I think, where you have shown percentage elemental composition.
This one, sir. This one.
Yeah.
Yeah.
So this, how this was calculated? Like antimony.
Normally SBSI, SBTI, and even SBSAI also I have prepared by taking the stoichiometric ratio, and thus I have taken the 50% of the sulfur, tin, and this one of the main element.
No, I thought because you might have done EDAX or HPS or something.
EDAX, I had done for tertiary element, this one as well as quaternary also.
So these values are from EDAX or theoretical values?
This is the theoretical, sir, whatever I have taken for the synthesis process.
No, but then you can try to measure from the EDAX.
Yes. Yes. Sir.
Okay. One more thing is like when you displace antimony with tin, how the charges are balanced?
How the
charges? Because antimony is trivalent, tin is divalent, or
yes.
So how those balances are satisfied?
How do you see that? Actually, I think polarization plays a great this one in this.
Because when you substitute one element with another, their charges they have to be same actually basically. Or there should be some mechanism by means of which that balance should take place.
But I'm not completely substituting, sir. I'm not completely substituting. I'm adding additional as an dopant.
Okay, you look into that in more detail. So thank you very much, Archana madam, for your nice presentation.
Organizers for giving me opportunity.
So,
sorry to interrupt you, sir, but Dr. Dipti Deshpande madam, our Director of Human Resources, Gokhale Education Society, has joined the session. Welcome, madam, for this session. We had a very informative session in the morning with Dr. Satya and Dr. Kulkarni, and now we are running the sessions in three parts. Two for biological sessions and one is the chemical session which you have joined. We have more than 150 participants for this conference. I once again welcome you, ma'am, for the conference.
Thank you. Thank you very much. Thank you.
can we proceed now?
Yes, sir. So Dr. Dilip Kumar Yadav, sir, is available, I think, sir.
Okay. So Dr. Dilip Kumar Yadav, are you there? Yadav, sir?
Dilip.
You are there, no?
Yes, sir. Dilip Kumar Yadav.
Yeah. Okay. So now you can present your work. So you have this presentation number 1. Efficient method for formation of carbon nitrogen bond moieties. And sir is from KJ Somai College with the
Actually, sir, I have a problem in my laptop, so I have sent already the mail to the. So
that is fine, but now can you present now? Is it possible?
Actually, sir, I have already sent the mail, and can you open from your side so that I can?
Yes, I am sharing. Yes, I am
okay,
sharing.
sir. Thank you, sir. Thank you, sir. So, good afternoon to all of you. Sorry for the inconvenience. Today I am going to present my research work, that is efficient method for the formation of carbon nitrogen bond moieties. Next slide, sir, please. So my scheme starts from the introduction. reaction scheme, result and discussion, and the finally acknowledgement. Next slide, sir. So, importance of the carbon-nitrogen bond moieties, as we all know that this is very important moieties which are present in various important parameters, fine and bulk chemical, polymer materials, and so on. Okay, sir. Next. Next. So these are some of the functional group which contain carbon-nitrogen bond. Out of this functional group, which we are going to focus on the imine part, that is carbon double bond nitrogen. Next slide, sir. So what is imine? Imines are highly versatile building blocks and having wide application in various fields such as pharmaceutical, agrochemicals, synthetic intermediates, and heterocycles. Next slide, sir. So what are the prior arts for the synthesis of imines? Condensation of amine with carbonyl compound, self-condensation of primary amine with oxidants, oxidation of secondary amines, and transition metal mediated hydrogen transfer from the secondary amines, direct reaction of nitroarine with primary alcohols, condensation of amine with the alcohol. So we are going to focus on this part, that is condensation of amine with alcohol. Imines are also synthesized by using different catalysts such as ruthenium, osmium, gold, palladium, platinum, copper, and iron. We are going to focus on the base mediated transition metal free synthesis of imine from alcohol and amine. Next slide, sir. So what are the disadvantages of the prior methods? They have used transition metals, expensive ligands or additives, longer reactions time, and use of organic solvents. Our approach is to use inexpensive and easily available base to develop ligand-free system, to develop solvent-free system, and it has a wider substrate applicability. Next slide, sir. This is my reaction scheme where we have to synthesize the imine by using N-phenyl urea and the aldehyde components in the presence of base at 135 degrees centigrade and 24 hours. Next slide, sir. So this is the optimization of my reaction condition. In the beginning, we have screened so many bases to check which base is giving the good yield, and it was found that potassium hydroxide and the cesium carbonate is giving the excellent yield of the corresponding imine. That is entry number from 1 to 8. Entry number 9 indicates in the absence of base we are not getting any product, and at the same time we have also optimized the base loading and the time factors. So entry number 11 indicates with decrease in time there is a decrease in the yield, and entry number 12C indicates that with decrease in the temperature there is a decrease in the yield of the product. Entry number 13, in entry number 13 we have used 99.99% of the potassium hydroxide just to check whether there is any contamination of the metal or not. Okay, so it gives an excellent yield of the product. So these are the optimized reaction parameters. N-phenyl urea 1 millimole, benzaldehyde 1.5 millimole, potassium hydroxide 0.2 millimole, temperature 135 degrees centigrade, and time is 24 hours. So with this optimized reaction parameters, we have studied so many substrates which are giving good to excellent yields with electron donating as well as the electron withdrawing group. We can see here reaction phi giving the less yield of the product because it is ortho substituted. It is might be due to the steric hindrance. Okay, next slide, sir. Next slide, sir. So again, we have studied different N-phenyl ureas with heterocycles as well as the ortho-para substituents. Electron donating groups are giving the good yields of the product, whereas the electron withdrawing groups are giving the less yield of the product. Next slide, sir. So this is the plausible reaction mechanism. N-phenylurea under the basic condition undergoes decomposition, gives you aniline, which reacts with the carbonyl compounds, and with elimination of the water molecules give me the final product, that is amine. Next slide, sir. Conclusion: we have developed efficient synthesis of amine from aldehyde and N-allylureas. It is metal-free and ligand-free condition to give the product. N-allylureas are stable as alternative to the amines. It has a general applicability. Next slide, sir. References, acknowledgement. Finally, I would like to acknowledge UGC, ICT, my guide, Professor Bhalchandra M. Mange, and the principal of KJ Somaiya College of Science and Commerce. Thank you. Any question?
Thank you, Dr. Yadav, for your nice presentation. Participants, any question? Okay, so Dr. Yadav, basically you have tried to avoid transmetal catalyst. So if you compare the yield and other things, comparative study have you done?
Comparative study with respect to what, sir? Transmetal free?
Yeah.
Actually, we have not compared as such, but we have tried to study the contamination, whether there is any contamination of the transmetal present in the bases. So for that, we have studied 99.99% of the potassium hydroxide, high purity potassium hydroxide. So it is actually, I can say that it is totally different with respect to the transmetal catalyzed reaction.
Okay, thank you. Any other question? Okay, so thank you very much. Thanks. Now we go to next presentation. That is OC 8. So, Mr. Vikrant S. Palekar, are you there?
Yes, sir.
Okay. So, this presentation number 8, and the title is Greener Approach for Aminolytic Depolymerization of Polyethylene Terephthalate Waste, and sir is from Arts, Commerce and Science College, Jawahar, Palka. So over to you, sir.
Very good afternoon, sir. Myself, Dr. Vikrant Palekar. I am from Gokhale Education Society, Arts, Commerce, Science College, Jawahar, Palka. So today I will be giving my presentation on the topic, that is greener approach for the amylotic depolymerization of polyethylene terephthalate waste. So introductory part, we can say that we are presently, we can say that we are in the polymer age, because nowadays polymer has changed dramatically in many aspects of human life. And nowadays we we can say that polymer have changed almost wood, glass and metal in almost all the fields, because of their wide applications in food, textiles, photographic fields, and for preparation of soft drink bottles. So why it is so prominently used? Because of its excellent properties, such as they are very lightweight, having very high strength, greater durability and longevity, and also they are very economically attractive and fabricate to use, and they are having low permeability of carbon, CO2. But we can see that as PET is not being hazardous polymer, but it's keep on accumulating inside the environment over the years, because of its very important property, that is it is not being non-biodegradable, and its ever growing production and waste generation is the biggest threat in front of the globe. So if we can see how it is, polyester is being prepared, as we can say it is a ester molecule. So it is being prepared by the condensation of terephthalic acid and ethylene glycol, and after it is being prepared, it is being can be used for the preparation of soft drink bottles, chips, apparels, and after all being used, it is being thrown inside the environment. So it is very necessary to address this particular issue. So as we can see in the prior art, there are n number of methods by which we can recycle this PET waste. So there are different ways. Physical recycling is one of the very promising approach, but it has a lot of advantages such as incineration, and so it can result into the various environmental issue. But in chemical recycling, here we can see in this particular slide, there are different ways by which we can convert this polymer into the monomeric unit. ways being used is methanolysis, where the polymeric ester again can be converted into ester. Then hydrolysis is one of the another approach. Then by using the ethylene glycol, we can convert this PET into the bis hydroxyethylene terephthalate. By using ammonia, we can convert into terephthalic amide. And by using different amines, we can again convert into. So chemical recycling is one of the very good approach by which we can convert this polymeric waste into the monomers. So by considering this, we have decided to concentrate our study on the aminolysis. As we can see that during the aminolysis, we can get very good monomeric unit, which has again advantages of being converted into the further useful chemicals. So this was our approach to concentrate on the aminolysis. For this, we are using the ionic liquid as a catalyst for the depolymerization of this PET. As we know that ionic liquids are the very versatilely used as a green solvent because of its promising properties, such as it is being completely containing the ions. It contains some cations and anions, and it is having very excellent properties such as it is being non-volatile, non-flammable, and soluble. So for that, we have choose imidazole as a cation, and we have tried to screen the different anions for that. So these are the different ionic liquids which are screened for the amylotic depolymerization of PET waste. Here you can see the ionic liquid being contained. The different 5 ionic liquids were screened. So we are having the different anions. Here you can see in the first way there is a Cl minus. In the second case, hexafluorophosphate, then tetrafluoroborate, then sulfuric, and this was the triflate. So these are the different ionic liquids were schemed. These are prepared by the reported methods which are available. So this is a scheme in which the PET is by using the hydrogen hydrate and by using the ionic liquid being converted into the terephthalic dihydrazide. So this is the monomer which we obtain after the reaction of PET with the hydrogen hydrate. Then the same approach has been used by just changing the amine. So for that we are using ethanol amine. So by again taking the PET and refluxing with the ethanol amine with the help of ionic liquid, here we can get another monomer. So that is a bis hydroxyethylene terephthalamide. So here you can see these two monomers are being promising moieties which again can be used for the further synthesis. So we are trying to check the influence of different ionic liquids on the yield of these monomers. So here we can see the serial number, that is the 4th ionic liquid, can give the promising yield of the both the monomers, that is terephthalic hydride and beta. So we have selected this particular triplet for the further studies. So here we can see in the next slide, we have also checked the effect of catalyst concentration on the reaction yield. So here we can say that 0.2 gram concentration can give excellent yield up to 85% or more up to 90% also of this both the monomers. Then also we have checked the effect of time on the yield. So here we can see almost 15 to 30 minutes are sufficient to get the yield of more than 80% of these monomers. So we are also again screen the effect of ratio of this ethanolamine and hydrogen hydrate. So we can see the 1:6 ratio is found to be more promising. So why higher ratio is required? As we are taking the polymer, so higher molar ratio is required for the this reagents. So then what are the products which are being obtained? They are again characterized by the different techniques such as IR. Here we can see the IR of the terephthalic hydrazide. This is NMR for the terephthalic dihydrazide, which are clearly showing the peaks for the NH and NH amines. So this is a IR for the second monomer, that is beta. This is a NMR which we got for the second monomer, that is beta. So in conclusion, we can say that aminolysis of PET waste can be effectively carried out by the greener catalysts such as ionic liquids, and the various different ionic liquids show very good catalytic activity for the amylolytic degradation of polymer waste. So the reaction required, time required for the reaction is very low, and we are giving very excellent use. And lastly, we can say what are the monomer obtained, which are obtained from the polymer waste, and they are having very large potential to be converted into further useful chemicals. So here we can say that we can effectively convert the polymeric waste into.
So thank you, sir, for your nice presentation. Why that particular one catalyst shows more efficiency?
Sir, that might be due to the acidic character of that particular anion, because in this particular case what we can see that the anion is a triplet, so it is being having very good acidic property. So that's why this particular cation anionic ionic liquid has shown the higher yield.
Okay, and you said that 1:6 is optimum one, but I can see that further increase in the ratio that gives up to 91% yield.
Yes, sir. That is that is correctly you are saying. But we can say the 1:6 ratio can be said as the optimum. If we increase the ratio, definitely we can get the higher yields also. But again, if we see the economical concern, the charges of the reagents also increases. So better to optimize that 1:6 ratio. We have decided.
Yeah. Okay, good. Yeah. Okay, all the best. Thank you very much. So
Thank you very
so
much,
now,
sir.
yeah, thank you. So
Thank
now
you
we
very much,
move.
sir.
Yeah, thank you. So now we move on to next presentation. That is the oral presentation number 9 by Wasim Beg. Wasim, are you there? Wasim? Okay, meanwhile, we can move ahead. Dr. C.B. Patil.
Hello. Good afternoon, sir.
Good afternoon. Okay. So I will introduce you quickly. So we have Dr. C.B. Patil from Department of Chemistry, Dr. P.R. Gregory Gogre College, Science College, Dule, and he is going to talk on epoxy biofiller, LDHS, nanocomposite preparation, thermal and mechanical properties. So over to you, sir.
Thank you, sir.
So you can start your presentation.
Yes, yes. Is my screen is visible to you, sir?
Yeah.
Am I audible to all of you?
You're audible.
Good afternoon, one and all. Honorable Professor Gajesh sir, chair of this session, and jury members of this session. Today, myself, Dr. C.B. Patil, is going to present my research work in this conference. First of all, I would like to extend my sincere thanks towards to the Mehta College for giving me an opportunity to present my research work over here. The topic of my talk is epoxy biofiller LDH nanocomposites, preparation, thermal and mechanical properties. As we all know, epoxy composites are one of the most widely used polymer composites. Epoxy-based composites materials have wide area of applications in automobile, aerospace, marine, and oil and gas industries because of their composite economy, good mechanical properties, high specific properties, and good adhesive properties, good heat resistance, and so on. For this reason, it's necessary to study and predict the mechanical and thermal behavior of epoxy-based composite materials. The prior regarding these epoxy composites suggests that there exists gap between research on the micro and nanoparticulate eggshell epoxy composite. This work is my present work is in efforts towards the utilization of West eggshell in the composite field. The research is present research is based on the fact that very limited or no study has been done on the eggshell nanoparticulate LDH epoxy alternately composites. In India, where there is a huge
presentation mode वर
Hello.
Go on presentation mode.
Okay.
Cup click करा तो खालचा. Yeah.
Okay. Are you able to view, sir?
Yeah.
Okay. The prior art of my research work suggests there exists gap between research on micro and nanoparticulate XL composites. This work is an effort towards the utilization of a waste eggshell in the composite field. The present research is based on the few facts that there is a very limited or no study has been done on XL nanoparticulate LDH epoxy composites. In India, there is a huge production of eggs daily. The research is done to find future research direction in the utilization of waste eggshell. The theme of this research is based on the value-added products from the waste eggshell. Here, in my research, we have prepared the biocalcium carbonate from the waste eggshell. We have recovered that biocalcium carbonate from eggshell with optimized and reported protocols, and then that eggshell calcium carbonate was characterized with XRD and TAME. Then that eggshell nanoparticles eggshell calcium carbonates was used to prepare the epoxy ternary composites with the LDH, that is layer double hydroxides. And that layer double hydroxide epoxy as well as biocalcium carbonate composites we have prepared with the help of slurry compounding methods. And after preparation of that composites were analyzed for their for their thermal and mechanical properties, as well as whether the formation of nanocomposite takes place or not. That we have find out with the help of X-ray diffraction, wide angle X-ray diffraction technique, and this wide angle results obtained on the wide angle X-ray diffraction where support that there will be the formation of nanocomposites, and there is which means as we increase the concentration of LDH by keeping concentration constant of biocalcium carbonate constant, with increasing concentration of LDH, there will be increase in the mechanical properties as well as thermal properties. There is significant, we found significant improvements in the mechanical properties. But as the concentration goes on increasing more than 5%, and then there will be the decrement in all mechanical properties. Here, that results are showing there is an increase in tensile strength, impact strength, and tensile modulus. Thermal properties also suggest there will be the increase in the thermal TGA results also supports there will be increase in the thermal stability of the composites. And with this, I'll have some conclusions. As the percentage of LDH in the epoxy matrix increases, tensile strength, elongation at break and impact strength increased and found maximum at 58% of LDH. After decrease due to the agglomeration, if we increase the concentration of nanoparticles or LDH, then there will be the chances of formation of agglomerates. And this improvement this improvement might be due to the mechanical properties were due to the load sharing of nanoparticles when subjected under load. And again, the thermal stability is also found was improved up to 58% of LDH sample as compared to the other compositions of nanocomposite and the neat epoxy because of the conduction of nanofiller. Thank you, sir.
Okay, thank you very much, sir. Participants, any question? Yes, so, sir, what kind of interaction is there between LDH and calcium carbonate?
Calcium, both means that both are LDH is a layer double hydroxide, and actually it is a layered material. The gap between these there are two layered plates is in nanometers. There is no any interaction between them, but they will means this is actually this is a ternary composition, sir. Means one is a platelet-like structure, and one is a spherical, and another third one is a polymer, sir.
Okay, and in XRD you get separate peaks for these two compositions.
Yes, sir.
So when we did something like a kind of physical mixing, you are saying?
Yes, a physical mixing, and that means if we increase the concentration of means only LDH, then we found that there is no that much improvement in the properties. Means only LDH with only LDH and epoxy, but we have added this LDH biocalcium carbonate and in epoxy that will found where significant improvement because of their ternary composition. Because here biocalcium carbonate is in spherical in nature, and that layer means LDH is in layer structure, sir. That's why might be their improvements are more as compared to the biocomponents as compared to the double complex epoxy and LDH composites, binary composite as compared to binary composite.
Okay, so thank you very much, Patil sir. Now we move on to next presentation. So, by chance, Wasim Beg is present now? If not, then we move on to next presentation by Dr. Jayasri Patil. Are you there, madam? Jayasri Patil? Jayasri, madam?
Dr. Jayashree Patil, madam.
Okay. If not, then Rasika Torne.
Yes, sir.
Okay, so let us go to next. That is by Dr. Rasika Torne. She's from Department of Chemistry, sir. Parsuram Bhau College, Pune. And the title of the talk is Proximate Analysis of Medicinally Important Ficus some difficult word it is. She will tell us what it is. So over to you, Madam Rasika.
Thank you, sir. One minute. Good afternoon to one and all. Welcome to International Virtual Conference, and it is honor for me to be a part of this international conference. I am Dr. Rasika Torni from Department of Chemistry, SP College, Pune. My topic is proximate analysis of medicinally important plant, that is Ficus bengalensis. This is abstract, and mainly I work on solvent extraction and loss on dry. Now first we'll see what is the importance of medicinal plant study. If we see in the world, 30% of the pharmaceutical preparations are manufactured from plants, and in Asia about 8,000 medicinal species are exist, and in India itself about 2,000 medicinal species are exist, but unfortunately only 6% have been explored for their biological activities, and only 15% have been explored phytochemically for their constituents. On this analogy of the above facts, research objectives have been planned. This is the plant on which I am working, and classification is given. In this classification, this plant belongs to family Moraceae, and it is authenticated in Botanical Survey of India, Pune. Its authentication is mentioned here. This is the authentication. Now, if you consider this particular plant, then almost all of the parts are used in medicine are used for medicinal purposes. Bark is used as astringent in the treatment of this particular diseases. Then leaves are used against dysentery and diarrhea. Fruits are used as tonic, and it is used against these diseases. Roots are used against this particular disease, and it is used as an medicine in the case of vomiting. So this plant material is collected from Baramati district, Pune, and particular aerial roots are used for the this study. This work is divided into two sections. Section 1 deals with the solvent extraction, and section 2 deals with loss on drying. First section, that is the solvent extraction, it is divided into two parts, that is cold solvent extraction and hot solvent extraction. So consider first cold solvent extraction. Cold solvent extraction means extraction is carried out at room temperature only, and therefore it is called as cold solvent extraction. This procedure is given over here. This plant material is extracted with different solvents, which is with non-polar to polar properties, and keeping that material for 24 hours at room temperature. Solvent was recovered, and that percentage yield was calculated. Results are depicted in table number 1. Next part, that is hot solvent extraction. difference between hot solvent and cold solvent is that cold solvent extraction is carried out at room temperature, while hot solvent extraction is carried out at boiling temperature of that particular solvent. So again, that procedure is given, and if you see here this apparatus, extraction is carried out in water bath. That solvent is removed under reduced pressure and crude extract is obtained, and that percentage yield was calculated. So see table 1 where core solvent extraction values, particular extractive values are given, and if we increase polarity, percentage goes on increasing. So for hexane it is non-polar, percentage yield is less as compared to methanol, where methanol is more polar and you will get more percentage of that particular extract. Next, this is for hot solvent extract. Extractive values are given for hot solvent extract. And see this value for hot solvent extract, still percentage yield is more as compared to cold solvent extract, because you are extracting that in water bath with boiling temperature of that particular solvent. So almost for almost all solvents, percentage yield is more as compared to that of cold solvent extract. Section 2 deals with the loss on drying method. This is very important method in natural product, because if we see, if you won't remove that moisture, then it enables maturation of harmful biological processes. So before starting any natural product study, it is
Please conclude.
advantageous to remove that moisture content. This LOD is also called as moisture content. And this particular method, that is you are using oven to dry that particular plant material. So advantage is that high reproducibility and reliable results are obtained, and as a result of this, it is the standard in many manufacturing process.
Madam, please conclude.
Okay, so this introduction is given, method is also given. You have to heat that at 550 degree, and here percentage of that particular loss on weight is given. In conclusion, hot solvent extract use high extractive values as compared to the cold solvent extract. In drying method, velocity and temperature of drying influences the quality and quantity of the ingredients present in that, and in future this plant material could be screened for various in vivo studies, and future plan is that to isolate, purify and characterize the active constituents responsible for the activity of these plants. These are the references which I have referred for my research study. Acknowledgement, special thanks for principal of SP College and head department of chemistry SP College Pune for their help and encouragement in this work. Thank you.
Thank you, madam. Any questions from the participants? Okay, madam, I think there are no questions for you from the participants. Just one query. What is common name of this particular?
Word. In Marathi it is called as word.
Okay, वडाचं झाड आहे ना तेच. मला हे वाटलं ते तसं.
Word. That is word. And it has many medicinal properties, but still some work is pending, and on that I am working.
Okay, thank you, madam. Thank you.
Okay, thank you, sir. Thank
So
you, sir.
now let us move on to next one. Is Dr. Pallavi Gawande there?
Yes, sir.
Okay. So Dr. Pallavi Gawande is going to give presentation on green synthesis of bimetallic gold silver nanoparticles from Moringa oleifera leaf extracts and its antimicrobial properties. So she is from Department of Forensic Chemistry and Toxicology, Government Institute of Forensic Science, Aurangabad. So over to you, madam. Yep, please start. Yes, we can see. We cannot hear you, madam.
Madam, you're not audible. Please unmute yourself.
Hello, sir. Hello.
Yes, yes, go ahead.
Good afternoon, everyone. I am Pallavi Gawande, student of MSc second year from Government Institute of Forensic Science, Aurangabad, and my topic of research is green synthesis of biometallic gold silver nanoparticle from Moringa oleifera leaf extract and its antimicrobial properties. So here, introduction, we will cover about introduction, objective, present work, methodology, result, conclusion, and references. So we all know about nanotechnology is an art of science of manipulating matter at the nanoseconds. Nanoparticles have a specific place in nanoscience and nanotechnology, not just in a view of their specific properties, but their decreased dimension. In the area of nanotechnology, metallic nanoparticle has shown number of properties and it has opened for numerous new pathways. So here are some objectives. The main objective of this study is to develop novel, efficient, convenient, selective, and environmentally benign synthetic method for nanoparticles which help the drug discovery and medicinal chemistry. The objective of the present study is to develop green methodologies for the synthesis of nanoparticles. Now, why green synthesis? As we know, green synthesis is easy and eco-friendly rather than physical and chemical method. It is efficient. It eliminate use of toxins and chemicals, consume less energy, and produce safer products and byproducts. Then we will ask about why Moringa oleifera only. So it has various different applications such as metal reducing agent, natural capping agent, non-toxic, eco-friendly, natural stabilizing agent, and colloidal. It has colloidal stability. Then bimetallic nanoparticle. Bimetallic nanoparticle have characteristic mixing pattern and geometrically architecture which enhances their functionality and function. They show better stability, selectivity, and catalytic activity over monometallic nanoparticles. So the present work, what we have done. So firstly, we prepared Moringa oleifera leaf extract, then synthesized the bimetallic nanoparticle. Characterization done by visible spectroscopy, XRD, FTR, and antimicrobial activity was performed. So here, our methodology was Moringa leaves were collected, washed with ionized water. As you can see here in a conical flask, there is a Moringa oleifera leaves, and there is a presence of deionized water. Then after that, leaves was allowed to air dry. Then moringa leaves was chopped. 50 gram of chopped moringa leaves was added in conical flask, and then 150 ml of ionized water was added in flask. Then conical flask kept in a hot plate at 60 degrees Celsius for 1 hour. Allowed to cool it. Then filter it with water number 42 filter paper. Then this way moringa oleifera leaf was extract was prepared. Then how we synthesize gold and silver nanoparticle. Then so firstly we prepared 1 millimolar of silver nitrate solution and 2 millimolar of chlororic acid solution prepared in 100 ml of deionized water. 20 ml of extract of moringa oleifera taken in round bottom flask. Then 60 ml of silver nitrate solution was added it in and kept in a magnetic stirrer for 15 to 30 minutes. As you can see here, color change was observed. Then 60 ml of chlororic acid solution was added at 1 minute interval. Then again kept on a magnetic stirrer for 30 minutes and allowed to cool it. After 24 hours, centrifuge at 1500 rpm for 5 minutes. As you can see, first the color is orangish, and then after completion, it appears like a black. So it happened due to when plant extract interact with the metal surface, then the charge on it reduces, so that this appears. Then when the centrifugation was completed and then supernatant was removed, sample was further used for analysis and characterization was done. Here the characterization of UV visible spectroscopy, you can see here on x-axis there is a wavelength and on y-axis there is a absorbance. The surface plasmon resonance of gold silver nanoparticle shows a peak centered near 354 nanometer was found for Moringa oleifera leaf extract, which correspond to the absorbance of Moringa, Moringa oleifera leaf extract of biometallic nanoparticles. Here you can see 354, which is most sharp.
Madam, please conclude.